Good morning, everyone. I want to welcome you to today's second annual Innovative Drug Discovery and Development Conference that was originally scheduled for 2020, but because of COVID had to be postponed. We're really grateful that the vast majority of our speakers agreed to join us again this year for this online venue, but we're really excited about the next couple of days. For those of you who aren't familiar with the Gulf Coast Consortia, we're really excited by what we've got going on here in the Houston, Galveston area, Texas Medical Center, as we've come together as a collaborative of what would traditionally be competing academic institutions to advance research and training programs that are much stronger when we work together, as opposed to what any one individual institution can accomplish by themselves. And so we're really happy and want to say thanks to all of our partner member and affiliate member institutions that you can see here. I want to give a big thanks to our Innovative Drug Discovery and Development Consortium Steering Committee. They are all absolutely fantastic and very experienced, incredible expertise across a broad range of drug development and even drug discovery, commercialization. I want to especially note out Stan Wadowich, who is the chair, and Cliff Steffen, who is the co-chair, as well as Peter Davies and Mike Mancini, who were co-founders of this years ago when it was called the Chemical Genomics Consortium. And to all of you, everybody participated in this, and you'll hear from many of them throughout the next 2 days as conveners and as chairs of some of the cores that will be highlighted in our showcase later this afternoon. I have to acknowledge CPRIT here because so much of what's happening in this area is because of them, because of direct funding. We couldn't do this without them. We're incredibly grateful for the amount of resources that they put into, academic institutions and small biotech companies for the purpose of developing and commercializing cancer therapeutics. I want to highlight here that we're going to hear from Cindy Walker-Peach, who heads their product development, and we're going to hear from her at 4:35, and that's going to be followed by our core facility networking showcase. And when I talk about our core facility networking showcase, here's just a brief overview of what some of these cores are that have been funded by CPRIT for various aspects of discovery and development and commercialization across the drug development pipeline. So we really hope that you'll join us later this afternoon. I believe those videos are up through the day, so you can take a look at them, and then you'll have the opportunity to jump into rooms where you can actually chat with these folks about their capacity and capabilities of their various cores. I do want to highlight the IDDD, that's our Innovative Drug Discovery and Development Consortium. We have a training program, and what that's looked like for the last couple of years has really been largely focused around our roundtable and workshop series, where we've looked at a number of drug development topics. We typically highlight some local cores as well as hear from kind of industry and build a network and learn about kind of best practices in both realms. And so that's going to continue. Along that line, we are going to take a break for the summer after June, but in June, we are going to have a second annual. For those of you that were able to join us last year, and I didn't change the date on this, this is June 3rd, 2021, so apologies for that. We heard from Alan Barrett and Roger Nozal, Alan from UTMB, the Seeley Institute for Vaccine Sciences, and Roger Nozal from Pfizer, who kind of gave us their opinions on looking forward at what SARS-CoV-2 vaccines were going to look like. And they've agreed to join us again on June 3rd, 2021, next month, to do kind of an update on prospects for the future. So we hope you'll join us for that as well. And then we have a lot of things we're really excited about for this training program. This is open to postdocs. We invite postdocs and graduate students from all of our institutions to participate, not only in the IDDD Roundtable Series, but we've got some things that are up and coming. We're planning a 2-week drug development and commercialization crash course for August, and diversity in drug development research course, which we're really excited about. We want to implement a postdoc industry happy hour to give some of our postdocs opportunities to network with folks in industry. We've got a lot more that we're working on, and so we hope that you'll just stay tuned for that and continue to participate with us in these things. And so I just want to kind of briefly highlight what we've got going on today. We're going to hear from our first keynote today at 9, Dr. Craig Lindsley from Vanderbilt, and then session 1 on discovery at 9:40. We have our first networking break at 11:10, and you'll notice that we have a few of these networking breaks. Really want to encourage you to utilize the Brella platform to set up meetings with folks that you might be interested in collaborating with. Brella has an algorithm to try to match at the top of your list of people, folks that would be somebody that you would recommend, that the algorithm would recommend that you should reach out to based on shared research interests. So take a look at that, and you should be able to set up a meeting directly through Brella. So we want everybody to take advantage of that. Even though we're not face-to-face, we hope that we can still have some networking going on. 11:25 is session 2 on advanced imaging for target validation and discovery. And then at 12:15, we have our poster session for our odd-numbered posters, another networking break and lunch at 12:55. At 1:30 is metabolism targeted therapeutics. We're really excited about this, as this is also highlighting one of the newer resources that we have in our IBT core. Session 4 on PK/PD and formulation at 3:15. And again, we're going to hear from CPRIT at 4:45, their update and company funding opportunities. And finally, for today, our core facility networking showcase at 5:15. And then we have another exciting day tomorrow. So with that, I want to turn this over to Dawn Koob, who is the one who has made all of the Brella conferencing platform possible for us. She's organized all the speakers, so I want to say a huge thanks to her, because without her, none of this would be possible.
Hello, everybody. Welcome to the imaging session for today. My name is Mike Mancini. I'm the director of one of the newer CPRIT resource grants, the Center for Advanced Microscopy and Image Informatics. And today, in a rather short session, we're going to run through a few talks that are connected directly to the center, which we nicknamed CAMI. CAMI supports all the projects that are being shown today and many others. I'm not going to go into a long intro for that. You can check out much more detail in the core sessions later today. Our first talk is by Fabio Sosi. Fabio, I met maybe 13, 14 years ago at a meeting in the mountains somewhere, and got to know him while he was still a student and postdoc, and not long thereafter we worked out an arrangement to bring him to Baylor, where he started learning and then eventually running the technical components, really the day-to-day of our integrated microscopy core at Baylor. And with that, I'll let Fabio take over, and we'll go from there.
Thank you, Mike, for the nice introduction, and thank you all for being here and listening to my presentation. As Mike hinted at, what I want to talk about is a project that has been going on for about 3 years or so in the CAMI group, and it's made of many parts and many moving parts of that. And most important ones are, of course, all the people that were involved. And I was maybe only the tip of the iceberg, but I have to thank especially Dr. Pankaj Singh and Arvind Rao for the informatics part, and Ragini Mistry, Hannah Johnson, Radhika Dandekar, and Maureen Mancini for all the experimental work that came into this project. Because what we wanted to tackle here was how to do quality control for single-cell imaging analytics. And that seems kind of a interesting idea, because you would imagine it was already all solved. But the problem is that when one look at nature, when you go from the macro level, so population level, from either human population or bacterial population, to the smallest possible biochemical reaction, for example, gene transcription, like shown here on the bottom right, then you can see that you have an enormous variation of any phenotype. And that's we can define as phenotypic heterogeneity, which is not due only to genetic variation, but to maybe intrinsic phenomena that are happening at any single chemical reaction. So why are we then so noisy as a system? Of course, I mean, the main reason could be simply because any reaction is non-100% efficient. So this, when you combine millions of reactions, all of them will create a certain amount of entropy and noise that will, cause this phenotypic variation. And if that was the case, and everything was stochastically and random, then it would obviously be not interesting in a lot of ways. But by studying, bacterial populations, for example, we can see that this gives a clear advantage in terms of evolutionary reasons, because you can actually think about it that when you have a drug treatment or you have a loss of nutrients, then what will happen is that maybe the whole bacterial colony will die. But all of it, no, some of them will survive. And so this gives them an advantage in terms of moving on with their colony repopulation and so on. So while this is happening, we asked some very basic questions. And these basic questions were really, can we quantify this variation between individuals? But more importantly, is it a reproducible phenomenon? And if so, can we exploit it in order to characterize certain perturbations? In our case, we're going to focus on chemicals and pollutants. Then basically what we want to do is to use a case scenario. And our case scenario is the estrogen receptor. And that is because Mike and I have studied the nuclear receptor family of transcription factors for a very long time. And so we are fairly knowledgeable about the mechanism of action. And what's important about the estrogen receptor is that it's a central gene expression regulator in many tissues, mostly productive and metabolic tissues. But for the purpose of this talk, what we are interested in is that it gets targeted by many natural and synthetic ligands, both drugs that are used in therapy, like tamoxifen, for example, for the aficionados in breast cancer universe, to endocrine disruptors like plasticizers. Bisphenol A is the prototype example, or pesticides, but also natural products. And the most famous ones are probably the isoflavones of soy or genistein. The reason why I'm bringing this up here is that every single molecule changes the receptor conformation in a specific manner that causes downstream effects that we can try to measure. And to give an example here, we can start looking at the estrogen receptor itself. The estrogen receptor is a transcription factor, lives in the nucleus. And so here we take 4 completely random fields of a 384-well plate that are immunolabeled for the estrogen receptor under basal conditions. So we didn't do anything to these cells. And you can probably appreciate that every cell has a different level of the estrogen receptor. And so what we, as a big group, decided to do is to semi-randomize our experimental conditions. Because what we thought is that let's see how human or cell influence would cause shifts in the distribution of the estrogen receptor in the population. And so we obviously took the same cell line. In this case, it's MCF7 breast cancer cells. And we used different passages, different thoughts, different batches of media. All the people involved did experiments, in their own days and whatever they wanted to do. We changed the vessel. How do we grow the cells? In multiple plates, on cover slips, and whatnot. We changed antibodies. So we would want to test if the antibody is what is causing this cell-to-cell variation in levels. We changed the way we imaged the cells. And that is because in the back of our mind, we were thinking, well, if somebody in Australia wants to repeat this experiment and they have no availability of the same microscopes we have, how does that influence what we can measure and what type of tools we can generate to help people out in terms of analyzing phenotypic heterogeneity? And also we changed image analysis software for the same particular reason. And here I'm showing you about 30 plus experiments. These are all independent biological replicates with several technical replicates, showing, in a heat map format, how the ER levels vary between different experiments. And so, Pankaj started thinking about how can we actually really confirm and validate if the distribution at the single cell level is actually a conserved metric. And so another way of saying it is that how do we define a bad experiment based on single cell distribution? And so we take the data, we Z-transform them, then he uses something that is used in signal processing called dynamic time warping. And he does that to the standard normal distribution. And then he calculates the Earth mover distance from from the experimental distribution to a reference distribution that I'm going to describe you in a second. And so if the metrics are bad, meaning that if this EMD is more than 3 standard deviations away from the reference experiment, then that experiment is considered a bad one, and we remove it from our analysis. And to do that in a formal way, what Pankaj did was to basically measure the pairwise distance between all these 30 plus experiments and generated the dendrogram. And in the dendrogram, and was important because we didn't know the ground truth of the distribution. Since it could have been completely random, we have no idea what the reference distribution would look like. And so we learned it from all our experiments because, as you hopefully can appreciate, there are really 3 different clusters that appear after hierarchical clustering. We have a very large one, which contains most of the experiments, and that's where we assume the ground truth exists. And then we have one experiment here called experiment 21, for the lack of a better name, that is a true outlier, lives alone in this dendrogram. And then we have 4 that also have their own little cluster. And so by the metric that I just described, all these 5 experiments are considered dubious, that we should either flag them as non-optimal or discard them because they didn't pass this first quality control level. But we were not satisfied. I mean, we're satisfied by seeing that actually the distribution was a quite reproducible feature, right? Because that means that we can actually use it for our quality control. But we are not also satisfied yet, and Pankaj started running in silico simulations that revealed that for this particular experiment, which is just measuring nuclear estrogen receptor levels, 5 independent biological replicates would be enough to have an approximate reference distribution based on what we may think of a ground truth. Now, as I said, this is specific on this endpoint, but it's applicable to any type of endpoint you are interested in. And then he simulated also the number of cells that are needed to have a faithful distribution that you can trust. And the result was approximately 1,500 cells per condition. And this, I hope, will help people that do single-cell analytics to kind of get a gauge of the number of acquisition that is needed, the number of cells that need to be acquired in order to have, fairly reproducible results. As I was hinting before, we can use known compounds that affect the estrogen receptor levels and see if we can measure a difference between the distributions under different conditions and create a second layer of quality control depending on a response, not just as a basal level of distribution. And these compounds are 17-beta-estradiol, or E2, which is the canonical agonist for the estrogen receptor that reduces the levels, for OHD or for hydroxytamoxifen, which is an estrogen receptor antagonist that stabilizes the receptor, and the fulvestrant or ICI, which is a selective estrogen receptor downregulator, which almost completely ablates the expression of the receptor. So again, Pankaj, with dynamic time warping, he can actually measure the distance of each compound through its own reference vehicle distribution, and that can be shown here. And that by measuring the EMD of all the treatments to the vehicle distribution, we can again qualify also a second level of quality control, which is the response level. And as you can probably see here with these dollar signs, we had 4 more experiments that had passed the first quality control layer, which was just comparing the vehicle basically, to a second quality control layer, which they fail, where one or more treatment didn't respond properly. Now, we don't know why that is. It could be because me, the operator, made a mistake and didn't add the compound at a particular day, or because, the ligand was maybe old, or for what other reason. But what we can say for sure is that those experiments have to be tagged as bad experiments and discarded from further analysis. So I'm going to tell you a test case linked to environmental toxicology. And we've done a lot of it, and I'm going to finish with that sort of overall arching picture. But I'm showing one particular example where we identified unique compounds using this method. And this is by testing 42 chemicals that are on the top 500 on the ATSDR registry in the United States EPA that are frequently found at Superfund sites across the United States. And why am I bringing this up is because Mike is part of a large grant, which is a Superfund grant, that funded us to develop high-throughput assays in order to characterize complex mixtures of environmental toxicants. And in order to do that, one has to start with references. And we tested over, almost now 200 reference compounds, and then also, lab-made mixtures in order to deconvolve more complex real-life situations. But anyways, here what I'm showing you is 3 heat maps of 3 features that we can extract from the single-cell data. And one is a classical one that you all understand very easily, which is the median level of the estrogen receptor. And here, the heat maps, you can read them by going basically more yellow you are, more hot you are, which means that you have higher levels of the receptor. And when you go into the black area, it means that the receptor is going down in this particular heat maps here, or it's at a lower level. So the median, of course, is the median level of the estrogen receptor in the nucleus in the cell population. QE is the quadratic entropy, which is a metric of entropy of the system. So it measures the variation of species, of ecological species in the population, if you will. And the third one is the Earth mover's distance, which is always a positive metric. That's why you see that largely is black. And that is because it's a distance from the reference distribution in the vehicle treated. Just by glancing at this, one can probably imagine that not a lot of compounds were a hit. We have only few that are easy to see here in the EMD. There's maybe about 8 or 9 out of 42, which was easy. What was interesting is that they fell into specific chemical classes. So we have polyaryl hydrocarbons, or PAHs, classical endocrine disruptors. We had metals, like mercury, cadmium, and chromium. We had very highly used pesticides that all changed the estrogen receptor levels. As you can see here, I'm showing for simplicity the median, and those are all reducing the estrogen receptor level. And a intermediate reagent, which is paracresol, which we didn't follow up on. So all I told you is actually kind of cool, I think. However, the fact that the receptor level is changing, it really does not tell us anything about activity. Because if you remember a few slides back, both an agonist and antagonist could have had the same exact effect, which is reducing the estrogen receptor. So estradiol, which is the agonist, was reducing it, but ICI, which is a full antagonist, was reducing it as well. And so this is maybe a convoluted way of telling you that no one assay is perfect. You always need orthogonal assays to complement the information so that one can have a clearer picture. And the way that we do it here is to include activity in our analysis, because that's what is missing from just measuring the levels. And so we are doing that by single molecule RNA fish, or SM fish, for ER target genes. What I'm going to show you today is the canonical one called GREB1, which is a highly stimulated estrogen receptor target. But this applies to any genes that you're interested in. And we're going to use single molecular RNA fish, which utilizes a panel on individual oligonucleotides that are all individually fluorescently tagged. And we're going to use 2 sets of them, just shown here in the middle. This is a DAPI-stained nucleus of MCF7 cells. If you can appreciate the little green dots, those are mature RNAs or steady-state RNAs. And those are basically, we can recognize them using an exon-specific set of oligonucleotides. And you can notice that also you have them in these larger blobs that are also labeled with a red probe, which is an intron set. And so this allows us to measure the active alleles in the cell. Now, MCF7 for GRB1 has 4 copies, as indicated here. And so we can measure both steady state and transcription bursting of the active alleles. So what happens with the compounds that I just showed you? And this is interesting because if we look at the stacked bar graph that highlights the fraction of cells in the population that have either 0, 1, 2, 3, or 4 active GRB1 alleles, one can see that, if we look at the PAHs, the benzoanthracene is quite active. It's not as strong or active as estradiol shown here, but it's actually quite active, while the benzofluorantine is really not that active as an agonist. Now, we're in the process of testing them as antagonists to see if they compete or if it competes off with estradiol, but by itself is really not active. In terms of the pesticides, again, the first DTT is actually very active. The disulfoton,
Fabio.
Yes, sir. I'm pretty much done. The disulfoton is also quite active, and the aziphosphomethyl is not that great. And the same thing can be said for the metals, where the cadmium is definitely the most active one. And so just to bring everything together, we can see here a heat map of EC50 values integrated between our assay, which are the bottom 3 panels here, with the Tkis high throughput assays for all the chemicals that we tested. And that's because the estrogen receptor is a central receptor in the Tkis library of assays, which is over 800 assays that were measured on over 4,000 compounds by the EPA and NIHS. But what is interesting that I want to point out is how our assay is showing a different set of results as compared to the Tkis. And this is in the left-hand side of the heat map are the active and gain compounds that were pretty much identified only by endogenous receptor measures. And this is what our assay is adding to the field of environmental toxicology. Now, some of them are obvious mishits, like cycloheximide, which is a protein translation inhibitor, which would be obviously a non-specific hit. But we identified some new ones, like I just showed you in terms of the pesticides, that we are going to be following up. And just to close, I want to thank the whole team, Mike and Pete first, because they believed in this project when probably no one else did, not even myself. And Dr. Pankaj Singh, because he really developed all the algorithms that are behind this type of analysis. And the rest of the committee team that obviously was part in doing these experiments, and obviously the funding that's always needed. And with that, I thank you all for your attention, and I'd be happy to answer any questions at the end of the session.
We're a little bit over questions. Let's try and save them for the end, and we'll just go into the next talk with Reid Powell. Reid, I met some years ago. He was a fresh grad student out of Texas A&M, located, in this case, in the Institute of Biosciences and Technology. Reid came through and grew into the high throughput screening center that has been set up now for a decade or over a decade, and graduated and stayed on, and now is a junior faculty member. And he's going to talk about his roles in high throughput imaging and informatics.
All right, thank you, Mike, for the nice introduction. And I also want to quickly thank everyone for taking the time out of the day, and as well as the organizers for putting this all together. And so, as Mike mentioned, my name's Reid Powell. I'm part of the Combinational Drug Discovery Program at Texas A&M, and I'll be talking about implementing deep neural network technologies across experimental scales. And so to preface this talk, I start with kind of a rhetorical test where if I was to give a panel of images and ask you all to put them in some sort of logical order, many of you would be able to put them in an order where you can group the different types of experiments together. And so that's kind of fascinated me, because in order to do that, you don't actually need that large of a context, but you're able to take general information such as, these are gray versus these are a different type of gray and these are green, and you're able to cluster them. Or you're able to take more of kind of a biologically focused knowledge where you can say, yes, these look more like a bright field image of organoids, while these are nuclear localized type signal, and these are more cytoplasmic or maybe some sort of cytoskeleton. And so that's really the core of at least part of my research program, where I've been working towards developing and implementing artificial intelligence systems that model human inference and then applying that to high throughput phenotypic screenings. And so the kind of main goals of why I'm doing that are really to shorten the assay development time by generating more generalizable analytical routines. Instead of developing a boutique type routine for every single assay, I'm working towards developing a single analytical system that can capture a very wide array of different types of screens, and then also to create enabling technologies to support these advanced cellular models, 3D models, and so forth, so that we can actually ask better questions in these high throughput screens. And so to do this, I'm using a technology known as deep neural networks. These are kind of a buzzword of the day. And so to quickly orient everyone on what they are, these are artificial neural networks, which are a machine learning approach that is inspired by the biological relay of information and the connection of neurons. They're then coupled with what's known as convolutional layers. And so those are layers that are responsible for actually learning features. And so unlike conventional computer vision, the feature extraction and feature engineering is coupled with the classification. And what's pretty much known about these deep neural networks is it requires a lot of data. They're always associated with big data. And in experimental science, we don't always have large data volumes. A lot of people will, depending on the scale of the research, they'll have smaller data sets, or you have to dynamically generate a data set. But you still need to be able to do quantification and actually make decisions on that data throughout the whole process. And so to do that and overcome those limitations, I'm using an approach known as transfer learning, which has two sub-approaches known as image embedding and fine-tuning. And so image embedding, you can take an unrelated data domain where you have a lot of information, such as cars and stop signs, and apply that and learn general features on how to understand and contextualize images, and then just use those feature spaces and then do what's known as image embedding or a linear projection of that information. As you then acquire more data, you can do fine-tuning where you're only training part of this feature learning step instead of the whole thing. And so both of those techniques can be used to overcome data limitations and apply these state-of-the-art technologies at different types of scales at different time points of your kind of experimental process. And so to exemplify this, I'm going to talk about one of our projects that we ran with a collaborator, Imad Shareki, who was at MD Anderson and is now at University of Michigan, where really the goal was to take patient-derived materials, in this case, patient-derived organoids that were embedded in a 3D matrix, Matrigel. They were plated and then treated with a panel of preclinical candidate drugs. And we were really trying to ask, which drugs should we take forward into patients? Which ones should we be more interested in? And to do that, we then looked at those over time. And so we just did simple bright field imaging over the course of 7 days. And then at the endpoint, we did a terminal cell titer glow to evaluate viability. And so this is kind of just a panel of the Z-projected organoids. You can see that they are making kind of these three-dimensional organoids. As again, this is a Z projection and a processing, and so they kind of look a little different than normal transmitted light. We then created a standardized data structuring algorithm where we do batch consolidation. We're taking all the information across the different experimental batches and different cell line models, consolidating them, and then splitting them into an 80-10-10 training, validation, and withheld set. And so we're using kind of these standard data practice for validation and modeling. We can then take a pre-existing model known as a ResNet 18. This is kind of one of the top models that's used in the deep learning field. And then early on in the experiment, we can use a pre-trained version of it and then do a linear embedding such as PCA analysis. And so that gives us kind of that immediate need of providing a quick quantification and a quick look at the data. As we then generate more data, we step back and we use what's known as auto machine learning. And so that's a machine learning, it's a deep neural network type infrastructure that is built from the known input and outputs, and then you can train and fine tune it as you're developing that data. And so to just show kind of how this works, I have, a 384 well plate where, where we've treated, in this case, 8 different drugs in 10-point dose response in quadruplicate. We can then visually assess how those look. And you can see that, with increasing drug concentrations, you lose organoids, and so you're mediating an effect. If you then look at a terminal endpoint, such as cell titer glow, you can see that there's different blocks where you have transitions from, in this case, right to left, from green to red, indicating that you have increased toxicity or some sort of change in viability. And then what's really kind of interesting is you can take those image embeddings and then do PCA analysis. And in the first principal component, you're actually able to resolve and get the same level of information you are from the cell titer glow. And so you're able to take a non-invasive, very cheap imaging modality and recapitulate the cell titer glow. And you have then an orthogonal readout. You know that the cell titer glow is actually mediating some sort of visual change. What we can also do is directly train these neural networks. And so instead of training a classification network, we're training regressive networks, so we can provide a continuous readout of response. And so shown here is how we're optimizing it. We're optimizing by loss instead of accuracy, which is typically done for classification. And so you can see in green and orange that it kind of goes down and they match each other. And so there's not overfitting. We can then use the withheld set, and you can see that we can get a nice linear regression. And so we could actually train a very nice model using the orthogonal readout. And so why would we want to do that? And the reason is because it creates a generalized tool that you can non-invasively track cells. It also can augment our interpretation of the endpoint data by back projecting the endpoints onto the dynamic read data. And so you're able to get, aspects of growth. And then you can also look at specific drugs, and you can see that there is a dose-dependent effect on growth. And so this is, again, predicted viability over time. You can see that it goes up and then begins to go down as you have increasing concentrations of dose in the colors. And you can evaluate and say that these are more cytosuppressive versus cytotoxic, shown by the slope of the line. And so just to quickly summarize, we're using pre-existing information where if we don't have a large data volume, we're using a lot of a prior information from different fields and performing embedding. And as we're generating data, we slowly shift off a prior information and create more data representation or representative data using fine-tuning and full training of networks. And then we're able to use orthogonal readouts as well as data structure to train these models. And that gives us an ability to create generalized tools as well as learn additional information and kind of turn a static endpoint into a more dynamic one using non-invasive technologies. And so with that, I would like to quickly acknowledge kind of our group, including our leadership, Peter Davies and Cliff Steffen, as well as the many people who actually do the work, do all the wet lab. All of our collaborators, including Imad Sharecki, who let me borrow the data, and CPRIT. And then I'll also be available in the combinational drug discovery programs session later today if you have any other additional questions. Thank you.
That's great, Reid. And again, hold the questions till the end of the next talk, which is by Vlad Sandaluce, or something close to that, pronunciation-wise. Vlad's a great example of one of the projects in our center that uses both microscopy and screening, and it's perfect for the surgeon scientist that he is as an assistant professor here at Baylor College of Medicine. Go ahead, Vlad.
Good morning. Thank you for the opportunity to talk this morning. So I'm going to go through about 6 years of work in about 8 minutes, just to make sure everybody's had sufficient coffee, and talk a little bit about cisplatin response and resistance. So first, I'd like to thank my collaborators, which are numerous, including the people that do the research in my lab and the lab of some of my collaborators like Wang-Ji Yu, Wu-Hao Lu, Sima Rawal, Mokim Kazi, Yun Chen, and Ying Henderson. And in particular for this talk, our collaborators in the high throughput drug screening cores and the imaging cores, specifically Cliff and Fabio's groups, and our funding sources, of course. Okay, so why talk about cisplatin? Well, we talk about cisplatin because as of 2019, we had 2 clinical trials which pretty definitively demonstrated that at least for head and neck cancer, which is the disease that I treat in the operating room, cisplatin is the gold standard systemic agent regardless of involvement of the human papillomavirus and development of the disease. So this is not a question of debate. This is the most proven drug that we have available for this particular disease. The problem with cisplatin was discovered when this guy was born, and it was introduced into clinical practice in the United States when this guy was born. This is me. This fellow predates me by about 130 years. So this is not exactly what you would consider to be a new drug. Despite this, the reality is that we don't know how to use cisplatin, at least in its optimal form. And for that, I offer the following proof, which is that as of 2021, the NRG cooperative group is planning to test high-dose cisplatin versus low-dose cisplatin in a prospective clinical trial of almost 1,000 patients. So clearly, if we knew how to use it, we wouldn't need to do a prospective clinical trial to figure that out. So what's the problem with cisplatin, and why is this even something that we need to study? Well, if you read the thousands of articles that look at cisplatin response and resistance, about half of them will tell you that cisplatin resistance correlates with EMT, or epithelial mesenchymal transition. A subset of those articles will tell you that EMT actually drives cisplatin resistance. And then in parallel, many hundreds of articles will tell you that EMT drives resistance to multiple other drugs. And then the other half of the thousands of articles will tell you that cisplatin resistance correlates with P-glycoprotein, that P-glycoprotein drives cisplatin resistance, and then of course that P-glycoprotein also drives resistance to multiple drugs. So that drives forward this conclusion, which is that cisplatin response and or resistance are non-specific. The problem with this conclusion is that it gives rise to a whole bunch of illogical conclusions. The first one is that whether you use cisplatin or another drug, it doesn't really matter. That once the tumor stops being responsive to cisplatin, it's unlikely to be responsive to another chemotherapeutic agent or targeted agent for that matter. That targeting intracellular ATP levels will fix the entire problem. And then especially relevant for head and neck cancer, that using induction chemotherapy using platinum agents is going to diminish the chemoradiation response on the back end, which is something for which we have empirical and prospective clinical trial data against. We use a slightly different approach, which is driven by 2 parts. So the first part is, if we think about what cisplatin needs to do in order for it to do its job of killing tumor cells, it has to bind DNA. So we think of a redox shield that literally surrounds the DNA, and that redox shield consists of reducing equivalents in the form of thiol-containing proteins such as glutathione and other primary reducing equivalents that are generated metabolically. The second part of this different paradigm is that the cell has to live in a balance between intracellular ROS and intracellular redox potential. And that balance is driven by carbon flux, which is used to regenerate these reducing equivalents. When you introduce exogenous ROS in the form of chemotherapy under radiation, you disturb that balance. If the cell is unable to equilibrate its reducing potential, then the cell dies. If the cell is able to rapidly shunt carbon flux and generate additional reducing equivalents, then it survives. So how do we study this paradigm? Well, we sort of break it down into 2 parts. And the first part is we need to study the shield, or the shield of reducing equivalents around the DNA. So the really painful way of doing that is using induction-coupled plasma mass spectroscopy, which is a very nice way to decide to play with hundreds of samples, all of which have to be generated using Aqua Regia, which if you haven't had the pleasure to work with, I highly recommend. It's a wonderful experience. That's sarcastic. And what we can rapidly, well, not so rapidly, what we can start to quantitatively understand is the intracellular dynamics of cisplatin in our cell line model. So for example, here we can easily see in a quantitative manner that most intracellular cisplatin does not actually bind the DNA, but exists within the cytosol and the other organelles outside of the nucleus. Using the same approach, we can actually demonstrate that cisplatin uptake in vitro is both time and dose dependent, and we can find a nearly linear relationship between cisplatin import and export and the dose, telling us that there's not very much energetics, at least in the initial phase of cisplatin import, and also that it takes cells several days to begin to export cisplatin or to begin to equilibrate to the initial insult. It is true that using this approach, we can actually demonstrate that there are some energetic constraints, which is something we have known and sort of supposed, at least for a period of time. So if we expose cells to a known concentration of cisplatin in the presence of a glycolytic inhibitor such as 2-deoxyglucose, we can see that we can double the intracellular platinum concentration in cell lines which are both sensitive and resistant to cisplatin, and we can definitely double the DNA-bound platinum concentration. So that tells us two things. It tells us that we can measure the actual amount of DNA that's bound, the actual amount of cisplatin that's bound to the DNA, and the energetics associated with that. Using the same approach, we can then measure the drivers of resistance and show, for example, that in cell lines which acquire cisplatin resistance, we can see a very dramatic decrease in DNA-bound platinum concentration. The high throughput approach is to use gamma H2AX foci, which, although they don't measure DNA-bound cisplatin, they give us a surrogate measure for the amount of damage that is caused essentially in real time by cisplatin. And this is work that we've been able to do in collaboration with Dr. Mancini's and Dr. Staggi's group, and essentially transition to a high throughput approach that allows us to look at the mean nuclear intensity of gamma H2AX foci and the fraction of positive cells. So what this allows us to do is to rapidly cycle through both measurements of endogenous cisplatin toxicity in a variety of cell lines, as well as looking at the acquisition of cisplatin resistance. So if we look at the cisplatin-resistant cell lines, for example, and we look at the fraction of positive cells and the mean nuclear intensity, what we can see is that although cell lines become resistant to cisplatin, they simply decrease their intracellular sensitivity to generation of gamma H2AX foci, but they don't lose the underlying mechanism of cell death. So hopefully these slides will project. They project fairly well on my screen, but the next step is to use the same high throughput imaging to study the intracellular localization of cisplatin. So if the cisplatin doesn't bind the DNA, doesn't get through the shield, where else does it go? And this is work that we've done in collaboration with the University of Pittsburgh, which has allowed us to study the intracellular localization of cisplatin in both cisplatin-sensitive and cisplatin-resistant cells and demonstrate that lysosomal trafficking accompanies the development of cisplatin resistance. So we match this high-throughput imaging approach with a secondary approach, which uses a multi-omics approach to study the metabolic balance that allows the redox shield to form. So this is based on a very simple premise, which is that reducing equivalents in the form of NADH and NADPH are able to absorb they're able to absorb intracellular ROS, and that the cell regenerates those reducing equivalents through differential carbon flux. And we have shown that there are multiple carbon pathways which can alter the rate of reducing equivalent formation. So if we take metabolomics, transcriptomics data, and we put it all together, what we can identify as those metabolic pathways which are essential for the acquisition of cisplatin resistance and for the differential response to cisplatin. And I'll finish here just to give you some idea of how all of this can come together. So if we take the depth map data, which is readily available, and we use a bioinformatics approach, what we can do is we can identify those genes that are essential for cells and those genes that are fairly inert or selective. If we then take our multi-omics approach using transcriptomic, metabolomic, and proteomic data, and we look at those genes that we know are regulated in the context of cisplatin resistance, and we put these two data sets together, what we can begin to identify for the first time is genes which, although are inert and completely irrelevant under normal growth conditions, become essential in the context of cisplatin exposure and acquired cisplatin resistance. And this allows us to begin to develop a transcriptional program that we think is both associated with and drives cisplatin response in head and neck cancer. And this is the model that we've developed using this multi-omics approach, basically a model that is fairly simple biochemically speaking, whereby If cisplatin is able to get through the redox shield, it'll bind the DNA and kill the cells. If cisplatin is absorbed by macromolecules such as proteins, it will not. And the way that cells are able to do that is to shunt metabolic activity in a way that is designed to increase macromolecule synthesis and essentially prevent the binding of cisplatin to the DNA. So this is a lot of data, but it really sort of speaks to the value of having both high throughput approaches and a multi-omics approach to ask and answer basic biological questions that, at least from our perspective, are very translationally relevant. Thank you.
Thank you.
Hi, everybody. Welcome back after what was, I'm sure, a brief but I hope pleasant lunch break. I'm Pete Davis, and it's my pleasure to serve as the moderator or facilitator for the third session in our conference, which is going to be focused on metabolism-targeted therapeutics. There's been a great deal of interest recently in the role that modification or alterations in cellular metabolism plays an important role in the phenotype of cancer cells, contributing to abnormalities in proliferation, metastasis, and particularly in the resistance to pharmacologic agents and different forms of therapy. So that there's a great deal of interest in understanding how we may be able to exploit these vulnerabilities in cancer cells that opens up new therapeutic opportunities. And that's really the focus of this afternoon's sessions. We're going to have 3 talks that are designed to cover various aspects of this problem. The first presentation by Mike Davies is really going to be a translational talk about the links between abnormal metabolism and the behavior of melanoma cells, particularly brain metastases. Then there's a second talk that will focus on the development of novel therapeutic agents that exploit metabolic vulnerabilities. And then the third talk will focus on new technologies for profiling and understanding the metabolism of cancer cells. So hopefully it's going to be an interesting and related set of presentations. It's my particular pleasure now to introduce the first speaker in this afternoon session, Dr. Michael Davies, and who is the professor and chairman of melanoma medical oncology at MD Anderson Cancer Center. Michael did his physician scientist training at UT Health Science Center in Houston, residency in the Mass General Hospital, and fellowship in MD Anderson. And Mike has really focused his research on a very interesting and clinically very important aspect of melanoma, and that is the issue of brain metastases and the particular strategies that may be used to treat metastatic melanoma in particular. And it's really my pleasure to turn the podium over to Mike. The title of his talk is The Role of Oxidative Phosphorylation in Metastasis and Therapeutic Resistance in Melanoma. So take it away, Mike.
All right. Thank you very much. Very unique opportunity to have your dad introduce you before you give a talk. Really happy that he didn't show some of my younger, more embarrassing photos as part of the introduction. So thank you very much to the organizers for the opportunity to speak today about our research. As Dr. Davis mentioned, again, I work on melanoma, which is the most aggressive form of cancer. And like other investigators, our work in the last few years has really highlighted the importance of metabolic pathways in this disease. And so happy to talk about that research today. Very briefly, these are my disclosures. And so melanoma really is the poster child of a disease that's been transformed by research. And it's really, to this point, been through 2 areas of investigation. So first has been studying the molecular heterogeneity and drivers of this disease, where the key feature of melanoma is that on average, melanomas have more mutations than any other cancer. And in the end, we can identify driver mutations in approximately 80% of our patients, which has led to the development of targeted therapies for this disease. What's been so exciting is to have in parallel breakthroughs in the understanding of the regulators of the anti-tumor immune response, particularly the development of checkpoint inhibitors by Jim Allison and others. And while it's great to make breakthroughs in understanding, what's been so exciting over the last decade is how this new information is translated into new therapies and markedly improved outcomes in patients. And so after decades of failure without a single agent showing a positive impact on overall survival in patients with stage 4 metastatic melanoma, from 2011 through 2020, we had 12 new regimens approved for this disease, all based on improving overall survival in our patients. And again, what's been so fascinating about this is that not a single one of them was a chemotherapy. It was really evenly divided between immune therapies that are taking the brakes off of the immune system, which we can use in all of our patients, or targeted therapies, particularly for the 50% of patients that have a BRAF V600 mutation that's present about 50% of our patients. And for those of you who are interested, most recently, we have our first combination regimen that combines immune and targeted therapies. And as a result, what we've seen over the last decade is really a quadrupling in both median and long-term survival rates in this disease, going from the one cancer that you really want to make sure you don't have to one of the most treatable cancers that's around. But that being said, we still have a lot of work to do. Now, again, these advances of immune therapy and targeted therapy have clearly demonstrated how important it is to understand the molecular biology of this disease and the immune regulators of this disease. But I think there's also growing evidence for the importance of cancer cell metabolism in this cancer and many others. And we've seen, again, that cancer cell metabolism is critically regulated by those oncogenic pathways that we're targeting with our targeted therapies, and growing evidence that cancer metabolism can also regulate the immune response and immune function, and importantly, has really sort of come to the forefront as an important driver of therapeutic resistance. And while for a long time we really focused on glycolysis and the Warburg effect, it's clear that there are many other pathways that are important, and I think many other therapeutic opportunities. So with that as a background, what I'd like to talk to you about today as an example is our studies on the role of oxidative phosphorylation in melanoma. First, to talk about its role in metastasis, and interestingly, a specific role in brain metastasis, which is one of the most lethal complications of this and many other cancers. And then OXPHOS as a therapeutic target and a strategy to overcome resistance to both immune and targeted therapies. And finally, briefly to lead into the discussion, some of our challenges and future directions. So as I mentioned, melanoma is the most aggressive form of skin cancer, and one of the most common and devastating complications of stage 4 distant metastatic disease is the development of brain metastases. Melanoma actually has a higher risk of brain metastasis than any of the other common solid tumors. And in contrast to other diseases, we often see this as an early event, with up to 20% of our patients having CNS involvement at the diagnosis of stage 4. And indeed, autopsy studies showed CNS involvement in over 80% of patients that die of this disease. In addition to being present initially, it's also a common site of treatment failure, often a solitary site of treatment failure for chemotherapy, biochemotherapy, and particularly targeted therapies as well. And historically, patients who've been diagnosed with brain metastasis from melanoma had a median survival of 4 months, by far the worst prognostic factor we have in this disease. And again, chemotherapies that cross the blood-brain barrier, dismal, really less than 10% of the time seeing any tumor shrinkage. And one of the things that we've tried to overcome as a challenge has been the practice throughout all cancers to exclude patients with brain metastases from participation in clinical trials, generally because of the concern of lack of efficacy of these agents. Initially blamed on the blood-brain barrier, but very clear once you can see a brain metastasis on an MRI, the blood-brain barrier has been disrupted. So many sort of historical perceptions that we've had to overcome to advance the treatment options for these patients. And that being said, we've made significant progress. So while patients with brain metastases were excluded from all of the phase 3 trials that led to the approval of the agents I showed you before, we now have several post-approval studies that have really demonstrated that our agents can have activity in brain metastases. Now, single-agent anti-PD-1 has a response rate in patients without brain mets of almost 40%, and in patients with brain mets that goes down to 20%, which was discouraging. But where we've seen much better activity is with combination immunotherapy. Our Ipi-Nivo combination therapy in patients with asymptomatic brain mets achieves clinical responses in the brain in approximately 50% of patients. So concerns about this being an immune privileged site really sort of overcome with this data. And what we've seen now is that over 90% of these responses are ongoing after several years. And so this overnight became a new standard approach for patients with asymptomatic brain mets. And while that represents significant progress, it's about 50-50 whether patients respond or don't respond. This is a toxic regimen where about 50% of patients get serious side effects. And we've seen recently that in patients who have symptomatic tumors, particularly those even needing just low doses of steroids, the response rate drops to less than 10%. In parallel, in the one clinical trial we've done with BRAF inhibitor plus MEK inhibitor targeted therapy in patients with a BRAF V600 mutation, we do see very good initial activity, response rates of 60%, and at least getting the disease control in over 80% of patients. No new toxicities observed in either of these studies. What was the key limitation with targeted therapy, though, is that on average, the responses in the brain only last 6 months. In contrast, responses in extracranial sites on average last a year. And in this trial, 50% of the patients progressed in the brain while their extracranial disease was still controlled. Now, it's possible this is because not enough drug is getting to the brain. And so we're trying trials with higher doses and with blood-brain barrier penetrating compounds. But there's also growing evidence that there's unique biology in the brain that may explain this differential activity. So we actually did one of the first studies looking at a small cohort of resected brain metastases from metastatic melanoma patients that were treated here at MD Anderson, where we had not only their brain metastasis tissue, but also tissue from other metastatic sites. They looked very similar in terms of their mutations, but when we did proteomic analysis, the striking finding was that in every single patient, the brain metastasis had hyperactivation of the PI3 kinase AKT oncogenic signaling pathway. And another group in Germany using immunohistochemistry found the same results. No difference in the MAP kinase pathway, but increase in the PI3 kinase AKT pathway. And to me, quite interesting because I did my PhD in a glioblastoma lab where one of the most common features we see in those tumors is loss of the tumor suppressor PTEN, which results in activation of the same pathway. Now, in our studies and the German study, we didn't really figure out what the mechanism of activation of the pathway was, but DYU here at MD Anderson showed that astrocytes in the brain can actually deliver microRNAs through exosomes to tumor cells to downregulate PTEN expression. Something I have to say is not necessarily always seen in clinical samples, but certainly, again, very compelling data in their Nature paper. And another group showed that cerebral spinal fluid can also activate that same PI3 kinase AKT pathway in vitro. And so together, these suggest that there really could be events that are happening specifically in the brain microenvironment. And so what we really wanted to do was to look for at these tumors globally to understand what their features are, their heterogeneity, and in particular, what are the differences between a brain metastasis and a non-CNS metastasis when I see a patient in my clinic, and how do I use that information to develop better treatments for the patient? And so Grant Fisher, a very talented MD PhD student at the time, now a pathology resident up in Boston, really helped drive this project. And so he and our pathologist went through all of the resected melanoma brain metastases that were in our tumor banks, and in the end, focusing on wanting to have tissue that we could do RNA-seq, and we can do this on FFPE. He ended up finding 88 melanoma brain metastases, or MBMs, from 76 patients. And then we went to our melanoma holdings for those patients and found 50 extracranial metastases, or ECMs, from 34 of those same patients. And while we did some DNA and some protein analyses, we really focused on the RNA-seq to get a global picture of these tumors. And so when we did unsupervised clustering, putting these tumors together, we found that they clustered by patient, not by metastatic site. And so we did not see global differences between the brain metastases and the extracranial metastases, but we did see about 500 genes that were significantly different between those two groups of tumors. And so when we used a variety of different pathway analyses, we found one consistent finding was that although we saw heterogeneity of the degree of immune infiltration among the brain metastases, and the degree of immune infiltration correlated with survival from craniotomy, what we saw consistently is that the brain metastases had less immune infiltrates than what we saw in the extracranial metastases for those patients. And this probably explains the decreased activity we see for anti-PD-1, which is really dependent on having those T cells present in the tumor microenvironment to generate a response. Now, on the molecular side, we looked at a variety of different things, but really what became the most striking difference between the brain mets and the extracranial mets was tumor metabolism. And very specifically, we saw this really striking upregulation of oxidative phosphorylation in the brain metastases from patients compared to their patient-matched extracranial metastases, and also compared to unmatched primary tumors. In melanoma, this OXPHOS phenotype seems to be primarily driven by a cofactor called PGC1 alpha. And so when we looked at those patients who had matching brain mets and extracranial mets, looked at the PGC1 alpha expression levels, we saw in about 3/4 of the patients higher expression of PGC1 alpha in the brain mets compared to the extracranial mets. Interestingly, this difference was perfectly recapitulated in preclinical models, and we looked at this in 2 different ways. So first, our collaborator, Dr. Sherry Holman at the Huntsman Cancer Institute, had a spontaneous brain metastasis model in immune-competent mouse where you induce the expression in melanocytes of the activating BRAF mutation, activation of the PI3 kinase pathway, and loss of P16. And these mice developed primary tumors, lung mets, and brain mets. And so she very generously sent us tumors from each of these sites. And when we did RNA-seq, we again found the highest levels of OXPHOS gene expression in the brain mets, which was higher than the lung mets, higher than the primary tumors, very similar to what we had seen in the patients. And then in our own lab, we took on the experiment of injecting human melanoma cells into mouse brain and mouse subcutaneous tissue. We did RNA-seq on those tissues and used the Xenome platform to specifically look at the human RNAs, and showed that in all of the cell lines, we saw this increase in OXPHOS in the brain metastases. And importantly, this came from direct injection. There was no sort of time for selection of which could metastasize there, but really seemed to be impact of the tumor microenvironment. And again, because we had a very controlled setting, we could also do direct metabolic profiling. So by both direct metabolite profiling and in vivo metabolic tracing of labeled glucose, we showed not only did we see increased TCA intermediates in the brain metastases, but very specifically through the metabolic tracing, this upregulation functionally of OXPHOS pathways with no change in glycolysis when we compared the brain mets to the sub-Q tumors. And so overall, very consistent findings saying that brain metastases had increased utilization of oxidative phosphorylation. Now, one of the things that's been also very interesting about this, and perhaps suspected based on the fact that we saw this with the direct injection of tumor cells, is that we're seeing these changes across multiple tumor types. And so Jason Hughes, the director of the MD Anderson Brain Tumor Bank, performed a very parallel study looking at matched brain mets and primary tumors or extracranial mets across multiple cancers, including breast cancer, non-small cell lung cancer, and renal cell carcinoma. And quite strikingly, in all of these cancers, we see increased OXPHOS in the tumors, increased PI3 kinase pathway activation, and decreased immune infiltrates, suggesting that there may be some true sort of universal features that we see being induced in the brain microenvironment, and in particular, suggest that OXPHOS could be a therapeutic target for brain metastases across different cancer types. Now, half of my lab works on brain metastases, the other half works on therapeutic resistance, and in particular, Vash Nanda, an assistant professor in my lab, has driven a lot of our work on resistance to targeted therapies, particularly those against the MAP kinase pathway. And Vash showed early on using RNA profiling that when we compared melanoma cell lines that underwent cell cycle arrest only versus apoptosis when we blocked the MAP kinase pathway, he saw that frequently the resistant cells were upregulating in a compensatory fashion oxidative phosphorylation, really all of those components of the electron transport chain, again, driven by PGC1 alpha specifically. And in our resistant cells, we saw this phenotype about half of the time, and strikingly, as shown in the upper right, when we looked at patient samples from patients who had progressed on BRAF or BRAF and MEK inhibitor targeted therapy regimens, where we had tissue from the time of progression to compare to the baseline, we similarly saw about half of the patients had this increased OXPHOS phenotype at the time of progression. Vash also generated acquired resistance models in the lab, and again, not every model, but about half the models, the acquired resistance models, would have this high OXPHOS phenotype. So here, as an example on the left, A375 is one of our sort of backbone models of BRAF mutant melanoma. When we grow the tumors in vivo, we can see with the yellow line that the tumors are markedly repressed by treatment with a MEK inhibitor, and we get stasis with an OXPHOS inhibitor. What was really interesting is that when Bosch isolated resistant clones with acquired resistance that now had a high OXPHOS phenotype, the A375R1 shown on the right, completely resistant to the MEK inhibitors, but now not only was the OXPHOS inhibitor effective, but it was more effective than in the parental cell line. So this adaptation to a high OXPHOS phenotype actually created a vulnerability where we now saw tumor regression with the OXPHOS inhibitor all by itself. And interestingly, didn't see any additive inhibition when we combined BRAF or MEK inhibitors with the OXPHOS inhibitor. So we were very interested to test that same OXPHOS inhibitor in our brain metastasis models. We started with, again, our intracranial injection of human melanoma cell lines into the brain. And this shows here the SK-MEL5 BRAF mutant melanoma cell line. It has high OXPHOS. It has de novo resistance to BRAF and MEK inhibitors. And surprising to us, despite the fact it has a BRAF mutation, treatment with the OXPHOS inhibitor alone, IXO10759, not only prolonged the survival of the mice, but actually cured almost a third of them as a single agent. So we then tested the drug in Sherry's spontaneous brain metastasis model, induced primary tumors, and then started treatment with the OXPHOS inhibitor. And these mice get very aggressive primary tumors. And interestingly, the OXPHOS inhibitor had no impact on primary tumor growth and did not therefore impact the survival of the mice. But when the mice died, we harvested different tissues and counted the metastases. And so shown to the right, the red bar is the vehicle, the blue bar is the OXPHOS inhibitor. In the mice, we saw a slight but non-significant inhibition of lung metastases with the OXPHOS inhibitor treatment. But in contrast, we saw absolutely zero brain metastases in the mice treated with the OXPHOS inhibitor. And we've again seen this preferential activity on brain metastases with Jason Hughes in both breast cancer and lung cancer models as well. And so, as I mentioned at the beginning, one of our challenges is patients with brain metastases frequently being excluded from clinical trials. Here we have potentially a new paradigm, a class of agents that may work best in patients with brain metastases, better than they work at other metastatic sites. Now, as I mentioned, we've worked on targeted therapy resistance in OXPHOS, but there's also growing evidence that OXPHOS can drive resistance to immune therapies. And I think some of the best work in the field is by Greg Delgoff and his colleagues at the University of Pittsburgh Medical Center. So they took a parallel approach of clinical and preclinical studies. So they obtained biopsies from metastatic melanoma patients who are about to undergo treatment with anti-PD-1 immunotherapy. And they put these tumor biopsies into the seahorse machine to assess both OXPHOS and glycolysis, and observed that only oxidative phosphorylation was associated with significant difference in outcomes. Patients with high OXPHOS were less likely to respond to immunotherapy, and if they did respond, their responses were shorter. And in parallel, he used a melanoma syngeneic model to develop subclones that had a hypermetabolic phenotype with high OXPHOS and high glycolysis that were resistant to anti-PD-1, and showed in those models only inhibition of OXPHOS would sensitize the tumors to anti-PD-1 treatment. Interestingly, didn't increase the degree of immune infiltration of the tumors, but improved the function of those infiltrating immune cells. And so we went back to our brain models to ask, do we see the same thing? And so we took the brain mets, again, the patient data, stratified them from highest to lowest OXPHOS based on an RNA-based signature. And we did see by immune score, which looks at hundreds of genes, that the high OXPHOS tumors had a lower immune score, but both by multiple sort of pathway analysis programs and by immunohistochemistry, OXPHOS did not correlate with degree of any immune cell infiltration, but did correlate with signatures of immune cell activation being decreased in the high OXPHOS tumors. And then similar to what Greg had done in his lab, he used a low-dose metformin regimen that seems to selectively inhibit OXPHOS in tumor cells, but not immune cells. We showed in our model of B16 brain metastases that the low-dose metformin treatment could again stimulate the immune response, and then had at least an additive effect in combination with anti-PD-1 in our brain metastasis models. Now, as I was sort of mentioning, well, again, the IX-10759 has been a great drug for us in the preclinical models as a direct and potent OXPHOS inhibitor. Unfortunately, its clinical development has been stopped due to toxicity. And so we've been looking for additional strategies using these models that we know are OXPHOS dependent. And in particular, our analyses of the brain metastases show that those high OXPHOS tumors not only had increased OXPHOS, but really looked like they were upregulating glutamine uptake and glutamine metabolism. And so we used glutaminase inhibitors and showed again in our brain metastasis models that even as single agents, that we could improve the survival of mice. And importantly, glutaminase inhibitors have been extremely well tolerated in patients. And so we're now looking at multiple different strategies, including looking at screening strategies to identify ways that we can target OXPHOS safely in patients to build upon and translate this preclinical work. So finally, just to summarize, again, I think melanoma is a really great example, but it sort of fits with the theme in cancer. Immune therapies and targeted therapies have had tremendous impact and have revolutionized cancer treatment and outcomes. But there's also growing evidence that in order to improve further, that cancer metabolism may become one of those additional pillars of anti-cancer therapy. And in particular, again, while there's been lots of work on glycolysis, I think oxidative phosphorylation appears to be very important, with evidence in multi-cancer sites that it can drive resistance to both immune and targeted therapies. And again, across all different cancer types, brain metastases remain one of our huge challenges. And so this finding of this selective upregulation and now Achilles heel with this dependence upon OXPHOS and brain mets does have, I think, significant implications to support drug development in this area. And that being said, we certainly have lots of challenges and questions in this space. So how can we safely target OXPHOS? And I would wonder, how can we selectively target OXPHOS in cancer cells compared to other cells? And certainly what we've learned from targeted therapy and immune therapy is, in addition to seeing what happens when we block a target, what are the mechanisms that cells try to use to compensate for blockade of that target, to mediate resistance and ultimately survival? And again, how do we look not only just at metabolic pathways, but at signaling pathways and immune pathways as well? So with that, I really would like to thank the many people who contributed to this work. We work with many different groups, both within MD Anderson and outside of it, and we're fortunate to have support from many different organizations. And so with that, I'd be happy to take any questions.
Thank you very much, Mike, for actually really a beautiful and very timely presentation in terms of doing it. It sets the stage for the other two talks that I think are going to follow. We have some questions on the Brella chat, and so let me present those two first. Prima Maleni is asking whether PI3 kinase AKT pathway inhibitors can be used to overcome MAP kinase inhibitor resistance.
Yeah, it's a really great question. And we, again, in our early studies, when we were looking not at RNA, but at the protein level, again, saw this upregulation of the PI3 kinase AKT pathway in brain mets. We also see that tumors with loss of PTEN have increased risk of brain metastasis. As you may know, targeting the PI3 kinase pathway has been one of the greatest challenges we've had in both drug development and clinical testing, that pathway is so centrally important that the challenge has been, how can we find a strategy to inhibit PI3 kinase pathways sufficiently without having unacceptable toxicity? So that idea of a therapeutic index. There are some isoform-specific PI3 kinase inhibitors that are now approved in the clinic, including alpha inhibitors, gamma inhibitors, and delta inhibitors. But it's still been a real challenge to really be able to knock down this pathway. And there have been one clinical trial of single agent PI3 kinase inhibitors with BKM120 in patients with brain mets. But again, our data shows that you only get this PI3 kinase activity in parallel to having activation of the MAP kinase pathway activity. So I think that, again, there's a very strong rationale for targeting the PI3 kinase pathway in brain mets. Our challenge is how can we do it effectively and safely, particularly in combination with other agents.
Thanks. And Catherine Shine was asking, do any of the TKIs have dual roles in inhibiting OXPHOS?
It's a really great. Yeah, no, it's a really great question. And what it sort of comes back to is what actually is driving the OXPHOS. In melanoma, it seems that OXPHOS is really driven at the level of a transcription factor called MITF. And so this now gets us to the questions of how can we actually regulate MITF. And in the work that Vaishnanda had published initially, he showed that it was critically regulated by mTOR, and that with mTOR we could cause nuclear export of MITF and use that as a strategy to block this OXPHOS phenotype. Unfortunately, OXPHOS inhibitors just as challenging as PI3 kinase inhibitors in terms of their toxicities. So RTK is clearly very much upstream at the cell surface of multiple pathways. I think this is where we start to get into the idea, and maybe we'll hear about this, of using screening programs as a way to test, sort of without any bias about what drugs can and can't inhibit OXPHOS, and potentially can we match that up with specific molecular profiles or specific cell types in order to develop translatable strategies. So I personally don't know specifically if the RTKs would do that or not, but it's a good question. Most of our work is focused pretty far downstream of those RTKs, though.
Thanks, Mike. And I actually have a question. You mentioned early on about both exosomes with RNA and CSF as potentially inducing that. Are there any indications that those pathways are working through PGC1, those extrinsic factors?
Yeah, no, so that is an experiment, at least in terms of the interaction with the astrocytes. That is an experiment and a grant that has yet to be funded. But we do not know yet whether that actually is mediating it or not.
And one other question was, and I think you may have addressed this, but any thoughts on what are the links between the activated OXPHOS and the suppression of infiltration by the cells? There's a lot of interest in the microenvironment, a suppressive microenvironment in tumors.
Yeah, it's interesting that, again, the data we have, and this is, again, an area that we're working on quite actively. In particular, we have a talented MD PhD student, Renato Guerreri, who's working on exactly what are the immune effects of OXPHOS in the tumor cells in the brain. Interestingly, to this point, the data by others and our early data really suggests that OXPHOS really impacts the activation status of the cells that are present in the brain mets. And it probably has to do with this competing for energy. If the tumor really has the ability to outcompete the immune cells for glucose, you essentially get starved immune cells, and that certainly suppresses their activity. But interesting to this point, it doesn't seem that OXPHOS necessarily explains the decrease in actual immune infiltrates that we see in the brain mets, that there may be other factors. And actually, the PI3 kinase pathway we've shown before may mediate that through actually driving VEGF expression. So it's a really interesting and unique microenvironment. Perhaps the closing thing I would say is just, again, for laboratory researchers, as much as we've often done our in vivo experiments with sub-Q models, there is this growing evidence that looking very specifically at different microenvironments, particularly those sites that are therapeutically challenging, brain is absolutely one, but I would argue bone and liver are the two other sites that are very hard for us to treat. Asking if really there are sort of site-specific pathways and dependencies is, I think, one of the other sort of hypotheses that comes out of our work.
Well, thank you very much, Mike. We should move on. I think if you transition to the Brella site, if people want to chat more with you about the work, you can do that in Brella, and the same for the audience, Mike, to do that. Thank you. Thank you very much.
Our next speaker is Dr. Hefferson, who is a professor of nanomedicine in Houston Methodist Academic Institute and leader of the Innovative Therapeutic Program in Houston Methodist Cancer Center. His PhD training is from MD Anderson and a postdoc from NCI, followed by quite a few years from the drug company. And back in the academia, his research interest is on delivery of therapeutic agents on target tissues, with particular emphasis on overcoming drug transport barriers. Dr. Sen.
Thank you very much, Dr. Liang. As Dr. Liang said, my title is Overcoming Transport Barriers in Cancer Treatment. So I'm going to show you one example of the drug transport barrier. Next slide, please. Well, here's a cartoon to show that there's a tumor inside a normal tissue. As you can see, the blood vessels in the tumor are completely different from the surrounding tissue. They are disorganized and they are not functional. That's why when you treat the patients through intravenous infusion of the drug molecules. Such molecules can reach the peripheral of the tumor, but cannot go inside. Next one, please. Here's one real-life example where we inoculate a tumor into the mammary gland of the mouse and put the live mouse under the fluorescent microscope. And if you expose the tumor, which is circled here, you can see the red blood cells. The red colors can reach the peripheral area of the red circle, the tumor area, but not deep inside the tumor. When we treat the mice with a fluorescent dye, these are macromolecular green fluorescence dyes that you can see. These dyes can again reach the peripheral of the tumor, but not deep inside the tumor tissue. That's why when you treat patients with highly potent drugs, such drugs may be able to kill the cancer cells very easily, very efficiently inside the petri dish, but they will not be able to kill the cancer cells in the live body because these drugs cannot see the cancer cells. The next slide, please. So back a few years ago, we listed the number of drug transport barriers and proposed ways to overcome such kind of barriers, at least in the case of the nanoparticulated drugs. We can see that once these drugs are injected into the body, first they need to survive the circulation because there are phagocytic organs, the liver, the spleen, for example. They need to escape these organs and then reach the tumor tissue. Once at the tumor tissue inside the microvessels, they need to pass the blood vessels and into the tumor cell area. However, in the process, they need to overcome the high interstitial fluid pressure there. Even once they have been uptaken by the tumor cells, then they still need to escape the endosomes, and they also cannot be pumped out by the drug efflux pump there. That's why when you design drug particles, you need to take all these factors into consideration and then enable the drug formulation to negotiate with all these transport barriers. The next slide, please. So over the years, we have developed several strategies to negotiate with the transport barriers, and we have successfully promoted several drug candidates into different stages of drug development, including the clinical trial. Here, I'm going to show you two strategies we have used. The first strategy is drug enrichment by affinity targeting. So when we took a look at the tumor tissue, we noticed that the most of the tumor tissues are inflamed. When these tumor cells and the stromal cells secrete the inflammatory cytokines, they promote the overexpression of a protein called E-selectin on the surface of the blood vessel. So a few years ago, we designed an affinity targeting molecule called the E-selectin cell aptamer. Here's a typo error there. This E-selectin cell aptamer can specifically bind to overexpress the E-selectin on the tumor blood vessel. The next slide, please. Here I will show the tissues next, the bone marrow tissues next to a metastatic tumor. as you can see that the blood vessels are stained a little bit lighter with the H&E staining. However, if you label them with if you probe them with the antibody, you can see they can be stained with green. Then once we use a red fluorescent E-selecting aptamer targeted the nanoparticle, you can see that as indicated with the yellow arrows, all these red particles will go to the green fluorescent blood vessel area. Once they are there, they are going to release drug cargoes and then reach to the tumor tissue. The next slide, please. And how does this strategy work? Here, I have one slide to show the blood vessels. On the left side is a blood vessel outside the bone marrow, and on the right side is a vessel inside the bone marrow with tumor metastasis. When these red nanoparticles are traveling in a normal blood vessel, they stay in the blood vessel. However, once they have reached the tumor area, the E-selecting aptamer actually bound to the E-selecting and then retain the nanoparticles there. Not only the nanoparticles retain there, but they actually escape. They have parted away from the fluorescent blood vessel. The next slide, please. Once they are in the inside the tissue, they are going to release the cargoes. I apologize for the confusion of the color. Now, on the right side, the nanoparticles are in green and the drug cargoes are in red. As you can see, next to the green nanoparticles, there were lots of red drug molecules. They just got released from the nanoparticle and permeate through the whole area to cover the whole area. Next slide, please. How does this strategy work? Well, one colleague in Weill Cornell Medical College in New York was interested in targeting the leukemia stem cells. And she found one compound called personalide, PTL in abbreviation. This PTL was able to kill the leukemia stem cells very, very efficiently in petri dish. However, when she was treating these animals, she needed to treat the mice 3 times a day and 7 days a week with limited accomplishment. Next slide, please. So when we packaged the same PTL into E-selecting targeted nanoparticle, we actually were able to only treat the mice once every 2 weeks. And after 2 treatments, we got very dramatic result on stem cell killing. The next slide, please. In this result, in this tumor engraftment assay, the PBS, in the PBS control, it shows that 20% engraftment, and there are the other control, the vehicle MSV control, or even we package the PTL into mice cells, it did not do anything. However, once we package the PTL into E-selecting cell aptamer conjugated drug molecules, we got a significant reduction on the percentage of engraftment, which reflects the leukemia stem cell activity. And here we have reduced the overall treatment dosage by 1,000-fold. The next slide, please. Another approach to overcome therapy in resistance, to overcome the therapeutic barrier is to use to harness the transporter properties of the immune cells, in this case, T cells. When we were working on the previous project, we noticed that certain nanoparticles can be uptaken by the dendritic cells inside the body. They not only stay in the dendritic cells, but also they promote the secretion of type 1 interferons. In this case, we have shown the overexpression of interferon beta and the downstream rentus expression from the dendritic cells. And mechanistically, these nanoparticles actually stimulate the TRIF and the MAVUS, leading to the activation of the type 1 interferon expression. The next slide, please. So we use that kind of nanoparticles to prepare therapeutic cancer vaccines, and then apply the vaccines to treat tumor-bearing mouse. Before that, we did several characterizations. One characterization was stimulation of dendritic cells. Now, dendritic cells are specific cells. Once they are activated, the cells they change the morphology and they develop a bunch of spikes there. In this case, we use CD11C driven the EGFP expression in the transgenic mouse. In the mouse control, then these animals were not treated with a nanoparticle. The CD11C positive cells were kind of roundish. On the right side, once we treated the mice with a cancer vaccine, you can see these cells develop lots of spikes, indicating their activation status. The next slide, please. In another study, we treated the tumor-bearing mice with vaccine inoculation in the pond, which is at the bottom of that. And then we labeled the vaccine particles with a copper 64 and use a PET-CT to monitor the migration of the vaccine particles. As you can see, at 3 hours, we see the signals in the popliteal lymph node at the bottom. And when the time goes on, the particles got transported upwards gradually into the tumor area and eventually reached multiple lymphatic organs throughout the body. The next slide, please. So when we applied this kind of vaccine particle to treat mice, we got excellent immune response and therapeutic efficacy. Here up there is an example of HER2 positive breast cancer. As you can see in the upper left side, these are untreated mice. You can see the number of brown stained CD3 positive T cells were kind of sparse. While after vaccination treatment, there were a lot more CD3 positive cells, indicating that we have pushed the T cells deeper into the tumor parenchyma. So we have evaluated therapeutic efficacy. We have demonstrated very effective inhibition of positive HER2 positive breast cancer in the primary tumor. And we have since then expanded our study. We have applied vaccines to treat metastatic breast cancer and treat colorectal cancer in murine models. In both cases, we have shown significant therapeutic efficacy over the control. In the case of colorectal cancer, we have also shown that our nanovaccine was a lot more potent than the poly-IC LC-based vaccine. I need to point out that the poly-IC LC-based vaccine has been used in human clinical trials, and this was the default choice currently. So I think we have a very good advantage in our nano vaccine in clinical trial. The next slide, please. So, well, over the time, we have also been optimizing our strategy to identify the nanoparticles that can be better phagocytosed by the dendritic cells. Next slide, please. We have also been screening for the vaccine compositions and also for genes that can modulate the vaccine transport. In this case, as you can see, the knockdown of the gene of interest can result to dramatic enrichment of the vaccine particles in the lymph node, indicating that this gene is negatively can negatively regulate vaccine activity. The next slide, please. Of course, curing tumor is just the first step of the overall process of managing human cancer. In this case, we have shown that with the therapeutic nano vaccine, we were able to eliminate the tumor nodule in the liver. However, the animals develop liver fibrosis. So we need to take this kind of effect into consideration. We not only need to cure the tumor, but also need to prevent the secondary event from happening. And the cancer treatment need to be combined with other treatment so that we can provide the best outcome. Next slide, please. Okay, I guess that's it. Thank you very much.
Thank you, Dr. Shen. I don't see any questions in the Barella. I guess people can still answer some questions if they have any other questions. I don't see any question at this point. We probably gonna go ahead close the session. I would like to thank you and thank other two speakers for this session, and looking forward to meet and then connect again in the following sessions. Thank you very much.
Hello, everybody, and welcome to day 2 of the second annual Gulf Coast Consortium Innovative Drug Discovery and Development Conference. I'm Phil Jones from MD Anderson. I've got the pleasure of hosting the first session of Tuesday here. So next up, we've got Alex Orteson from Vanderbilt Cancer Center in Nashville. Alex is a research associate professor in pharmacology and chemistry there at Vanderbilt. He did his PhD at Emory. He then went to Colorado State University and from there moved on to GSK, where he made a significant impact on a number of GSK's kinase programs looking at covalent inhibition and was involved in the BRAF inhibited BRAFINIB. Like Rima, he stepped away from pharma in 2008 and then moved across to Vanderbilt, where he's been ever since, working closely together with Dr. Stephen Fezzik around fragment-based drug discovery and has been successful on a number of projects, epigenetic therapies, and specifically around on the whole K-Ras field. And he's here to tell us about some of that today. So, Alex, over to you.
All right, thanks, Phil. As Phil mentioned, I'm going to tell you a lot about what Vanderbilt has done in the RAS field, and I'll point out right up front that much of this was done on Vanderbilt's campus, which you can see on my slide. A significant part of the work was either done by or in collaboration with Boehringer Ingelheim in their Vienna labs. So those of you that are in oncology probably need very, very little introduction to K-Ras and the Ras pathway in general. But for those of you that are not, here's a very quick primer. So you need to know that Ras is a conformational switch. It has a conformational change that it undergoes when it exchanges binding to an inactive GDP-bound form to an active GTP-bound form, and this happens with the help of exchange factor proteins. And in its active state, it lies at the top of a tremendous number of signaling cascades, only a portion of which are shown here. And it turns out that these signaling cascades actually control a great number of processes in your cells, many of which are related to growth and proliferation. And so the consequences of this breaking are pretty severe. And of course, RAS, quite famously, is mutated in as much as 30% of all human cancers. Also, very famously, RAS has been very intractable to inhibition historically, up until very recently. Many drugs upstream of RAS and many drugs that hit nodes downstream of RAS have really failed to deliver patient benefit. And starting about 10 years ago, there was a bit of a renaissance in RAS research to kind of go again right at the heart of the matter, so to speak. And our lab was a part of that renaissance. And I want to tell you a bit about what we've done both for direct binders to RAS as well as indirect binding to indirect inhibition of RAS via binding to SOS. And so to understand our projects, you do have to understand a little bit of the technology that we use. So Phil mentioned that I've been closely associated with the Fezzik lab, and Professor Fezzik is very dedicated to the concept of NMR-based fragment screening to discover starting points. If you're unfamiliar with the technique, again, a small primer is that every protein has a two-dimensional fingerprint that reads out the specific shifts of individual amide NHs in a two-dimensional way. And a shift in those locations of those shifts, so shown here from the green going to white, is indicative of some perturbation of the electronic or steric character around one of those NHs, such as when a small molecular weight fragment binds. And the concept behind SAR by NMR is that you find one of these weak binders, you optimize it a little bit, conduct a second site screen to find something that binds nearby, and then systematically build up a drug little by little. And in fact, we applied this direct concept to RAS some 10 years or so ago, screening both for GDP-bound RAS and GTP-bound RAS. And a little bit surprisingly, we found a fair number of hits, most of which bind to both forms, although a few of the compounds had some leanings of selectivity in one direction or the other. But the real key here is that we were able to crystallize several of these hits to a rather unknown pocket on RAS in its GTP-bound form. And you can see that pocket here with one of these small molecules. And of course, we engaged in a little bit of structure-based design to extend those molecules down the RAS binding cleft and improve the rather weak potency. But even the improved molecules for the GDP-bound form of RAS remained fairly weak at this stage of the project, really only getting to the few hundreds of micromolar binding. And because we were binding the inactive form of RAS, any functional output really had everything to do with whether or not they were inhibiting the activation of RAS as measured by exchange of nucleotide. And they did, but again, rather weakly. And so it was at this point that we actually partnered with Boehringer Ingelheim and sort of put heads together, combining our fragment hits, their fragment hits, our expertise, their expertise, and finally cracked by Dirk Kessler and his team at BI, the first co-crystal structure is a molecule bound to GTP, the active form of RAS. And this, of course, initiated a number of optimization activities. A very clever team of medicinal chemists there attached on this spirocycle that built in a new interaction to a threonine out here and a glutamate, bringing the affinity down into the 30 micromolar range, both as measured by NMR, as well as a FRET assay that assesses disruption of K-Ras and SOS interactions. They then continue to elaborate on that molecule, iteratively building up to a tool molecule BI-2852, as shown here, which is believed to be the first sub-micromolar reversible binder to K-Ras. Now, interestingly enough, this compound has a bit of a pan-RAS profile. It binds to all isoforms of RAS, all mutants of RAS that we know of, with a relatively similar affinity. And if you're interested in playing around with this yourself, BI has made it available on their collaboration platform, OpenMe. Let me show you a little bit about what they have published and known about it. It's a quite potent inhibitor of the protein-protein interactions driven by the active form of RAS. As seen here, it interrupts CRAF interactions as well as PI3K interactions with RAS. And accordingly, it's actually a reasonable inhibitor of cellular response. It inhibits phospho-ERK signals downstream of RAS and inhibits proliferation at low micromolar concentrations, and shows a pretty reasonable window over a structurally matched negative control that is only different by this stereocenter. So a pretty interesting molecule, and as I said, probably the first sub-micromolar reversible binder to RAS. But one thing that both of these chemotypes have in common, these indoles that I've shown you, is they still bind to the primary binding site that we uncovered in the original fragment screen. And this pocket is actually created by the rotation of tyrosine 71 out of the way. Now, when it rotates, it carries more protein with it, including this methionine, opening up a second site cleft. And we looked at that second site cleft as an opportunity to find a second site binder that we could latch onto the first site binders and then build up our affinity per the SAR by NMR paradigm. However, that's not as easy as it sounds sometimes. When we had our first generation compounds in hand, they were so weak that we really could not saturate the first site in the way that we had wanted to. So we had to come up with a different strategy. And our strategy was this. We thought instead of reversibly saturating the first site, maybe we would irreversibly saturate it by creating an artificial protein and a modified version of it that would covalently block that first site, allowing us to conduct the second site screen. So we looked really closely at the protein and identified 6 residues around that primary binding site, mutated each one of those to a cysteine residue in turn, took each one of those cysteine mutants and reacted it with a group of thiophiles that would covalently latch on, analyzed each of these now modified proteins by mass spec to make sure we had actually attached the small molecule, looked at them by NMR, looked at them by X-ray crystallography, etc., eventually convincing ourselves that this residue here, serine 39, when mutated to a cysteine and attached to this thiol-containing benzimidazole, perfectly recapitulated the binding mode of the reversible binders, but with an irreversible molecule blocked in. So there was our magic bullet. That's what we wanted for a blocked first site. We ran our second site screen using our fragment library, and again, we found hits with a much different shifting pattern. Now, I can't yet tell you exactly what these hits look like. There's still some protection around those at this stage. However, what I can say is that we were able to co-crystallize these molecules with the modified protein. And we got a bit of a surprise. These molecules did not bind adjacent to the first site as we expected. The secondary cleft would have been here in between the mobile switch 1 and switch 2 regions. Instead, they bound actually underneath this chunk of the protein in this pocket kind of highlighted in blue. Now, this was a little bit disappointing because we couldn't link them to our first site. We would actually have to tunnel through the protein to do that, which is quite impossible. However, at the same time we were doing these experiments, Kayvon Shokat and his team at UCSF had uncovered covalent modifiers of the G12C mutant of K-Ras. Those compounds actually access that same pocket. So now we had a bit of an idea. Maybe we could utilize these hits for something similar. And just to make sure you understand the differences in technique, Shokat and his team had used fragment tethering in which they intentionally looked at electrophilic compounds that would latch on to a pre-existing cysteine in the molecule, and then systematically optimized them for reactivity, optimized them further for potency, and have created very, very high-quality molecules. This technique was used even to create the first clinical RAS inhibitors that you've probably heard about from Amgen and Mirati. And in fact, the Amgen molecule may be approved, maybe the first K-Ras molecule approved for patient use ever, which is pretty exciting. Our technique is a little bit different in that we created an artificial protein to find a reversible binder. And our concept was then to optimize that reversible binding and then and only then reach out to attach it covalently and create a new G12C inhibitor. And this actually worked rather well. And again, I can't talk about all the details of the G12C inhibitor that we have in hand, but it is an amazingly potent inhibitor of K-Ras-driven protein-protein interactions, a sub-10 nanomolar inhibitor of pathway inhibition as measured by phospho-ERK, and a very potent inhibitor of cell proliferation. Even more exciting, optimized versions of this are suitable for use in vivo and then show nice dose-responsive tumor growth inhibition and even regression at the highest doses. This inhibition of tumor growth is, of course, associated with a very nice PD response. You can get a downstream marker. And at the highest doses, you can actually see evidence of apoptosis in the tumors. So a very nice asset. And what I've shown you with my direct binding sort of RAS unit here is that we took an original set of fragment screens that identified hits in 2 different sites. And with proper optimization and tweaking, we were able to create not only a G12C inhibitor, as others have done, but also inhibitors that address the other RAS mutants that are important in a number of cancers beyond G12C. So stay tuned for more details as some of these begin to roll out. But I want to stop the RAS direct story there and move on to our indirect attempts now, in which we really were looking at binding to SOS as an indirect way of getting into RAS. And I'll confess that we stumbled into this quite by accident, so this requires a little bit of background. The indole-based direct RAS binders that I've shown you before have the same effect of inhibiting the activation of RAS. They inhibit nucleotide exchange on RAS. And you're seeing right here some of the raw output of this type of assay. So this is basically a rate curve where a non-fluorescent nucleotide goes in, popping out a fluorescently labeled nucleotide and leading to a decrease in signal. Inhibitor would flatten out that rate curve. And this is shown very nicely with the BI tool 2852, where you've got a nice dose-responsive flattening of the curve. But at the same time we were working on these, we uncovered a somewhat related chemical template that had exactly the opposite effect. It actually steepened these nucleotide exchange curves, effectively activating RAS, which is unexpected and a little bit weird. However, they do this at pretty potent EC50s, around 10 micromolar in the best cases. So we took a poke at them in cells and verified that indeed this activation leads to higher levels of RAS GTP in a cell, and you can watch this grow up over time. And so then you would expect higher levels of phospho-ERK and phospho-AKT downstream of RAS. But that's not quite what we see, and here's where this gets very interesting. So you're looking at a lot of Western blots here that go from an inactive control molecule down to a more active activator of RAS. And at low concentrations for phospho-ERK, you do see the expected increase in activated ERK. But each one of these molecules then kicks over to inhibition of that downstream signaling in a way that appears to be correlated with nucleotide exchange activity. Even more intriguingly, phospho-AKT downstream of RAS goes straight down, direct inhibition, which is really intriguing. And it turns out this is functionally useful because these compounds are actually rather good at killing cells, that are cancer cells, in a selective way, at least as compared to the negative control. So we began to hypothesize that we had stumbled into a new way of getting at RAS inhibition. And of course, the reason for this is that these molecules bind SOS. And again, we've reported this, so I won't go through it in a hurry. But you're looking at here at a ternary co-crystal structure of the catalytic domain of SOS in gold, with a known allosteric binding location for RAS on one end, and apo-RAS under the process of having nucleotide exchange happen on this end. And sure enough, there's our activator compounds in green that are binding to another allosteric pocket directly adjacent to the business end of RAS. And this pocket is actually quite attractive. It's got a hydrophobic base, some polar residues around the periphery, and we liked everything about it except the way these molecules bound into it, with this rather unusual U shape that probably has the chemists in the audience shuddering a little bit. So with this knowledge in hand, we thought we would find additional chemical matter that bound to this pocket and see if we could recapitulate the pharmacology. So we turned to our old friend NMR-based fragment screening for this purpose first. However, we couldn't quite do that on SOS because frankly, the molecule's too big. We would have a huge diffuse set of shifts that we couldn't interpret. So instead of N15 labeling the protein, we C13 labeled it using only a selected set of residues that turn out to be spread across the catalytic domain of SOS, as you can see here in magenta. Using a combination of reported assignments as well as some in-house work, we confidently labeled every signal that we could see and convinced ourselves that we could read out for binding to SOS using this C13-based screening platform. So we ran our fragment library, and as fragment libraries do, we found some binders, a small smattering of which are shown here. Now, most of the binders that we identified bound to that activator site, kind of as expected. There were some others there, but we were actually amazingly successful at co-crystallizing these molecules with SOS in that activator site, and that opened our door to being able to do a few more things with these molecules. And so I'm going to show you now two of these hits as one part of the story. These are fairly weak molecules, as fragments usually are, and they bind quite differently in the activator site. So, for example, this phenylsulfonamide puts a phenyl ring kind of deep into the hydrophobic pocket, makes some polar interactions with an asparagine and a histidine kind of at the lip. But what's really interesting is this benzyl imidazole binds this pocket in a quite different way. It actually projects this phenyl ring underneath a phenylalanine residue that is now rotated up and away from the pocket, creating this lipophilic space. And so we reasoned that we could actually start combining features of these fragments and a merging strategy to create better binders. And that's exactly what we did. taking this phenyl ring from the benzoimidazole, latching it onto our most prevalent series that hits these phenyl sulfonamides, got us down below 100 micromolar in binding affinity and forced that Phe flip even when it wasn't native to do so. So now we were off to the races. We used a little bit more structure-based design to increase pocket occupation under that phenylalanine, to reach out and engage a water-mediated network around this aspartate 887. And we got down to single-digit micromolar binders that were quite potent exchangers in the nucleotide exchange assay. And indeed, these had the exact same phospho-ERK response that we had seen with our accidentally discovered activators, indicating that this might even be the native activity of this pocket. So that was strategy number one to get new chemical matter. The second strategy was using the nucleotide exchange assay itself to perform a high-throughput screen. And so we did that thinking that we would find both activators and inhibitors. But a screen of 150,000 compounds in the Vanderbilt collection actually revealed only activator compounds, again, a smattering of which are shown here. Now some are pretty ugly. Some look like dopamine receptor antagonists, but there were a handful of molecules that looked reasonably attractive as the potential for a tool molecule or even a drug. And we set about crystallizing these with sauce to see if we could do anything about them. And interestingly enough, again, they all bound in that Phe up binding mode. Now, I don't have time to tell you about all the medchem that was done down there. We've published relatively extensively in this, but I do want to tell you one story centered on this hit because it bears a lot of resemblance to the hit that we also saw in the fragment-based screen. So taking this tricyclic benzimidazole, we actually pared it down and did a little bit of standard medchem and came up with a molecule that looks like this without that third ring that activates exchange at about 10 micromolar. And its co-crystal structure reveals more or less what we thought. This benzyl piece sticks under the phenylalanine in that Phe up binding mode. The NH2 occupies a water-mediated network with aspartate 887. And those two facets provided the clue to additional optimization. So we popped out the water molecule by replacing this with a piperazine that now makes a direct interaction with aspartate 887. We fattened up the lipophilic matter up underneath phenylalanine 890, and we tweaked the sort of back pocket substitution with this monochloro. And the result of that was some really, really efficient activators. In fact, they were so good at activating nucleotide exchange that our assay had a really tough time measuring them. So we abandoned our nucleotide exchange assay, moved to a binding assay that we set up in the FPA competitive assay format, looking at displacement of a labeled probe. We found that our best-in-class molecules bound at about half a micromolar, and then we used this assay to engender some additional optimizations. Now, we ultimately got to molecules that look like this guy. Again, reinforcing that interaction with aspartate 887 with this spirocyclic guy, increasing our pocket occupation by substituting at the 4 position just right to sort of project down chemical matter in, got us down to molecules that were single-digit nanomolar binders to sauce and had really, really potent effects in cells of that same sort of ERK activation and then kickover to inhibition effect as I showed you before. Now, again, these tool molecules were good for a lot of things. We've done a lot of science with them. I don't have time to tell you all of the science. What I do want to tell you is that we were able to uncover what was going on with this crazy up and down biphasic ERK effect. And so I'll show you that little bit of science here before I move on. Binding of the small molecules to sauce is quite well established at this point through multiple chemical series that it leads to increases in activated Ras, which then lead to increases in the MAPK pathway downstream. Now, phosphorylated ERK is also more active ERK, and it turns out that sauce can be a substrate of this active ERK. So ERK goes up and phosphorylates sauce at a very specific residue, and that phosphorylation serves to break up the interaction of sauce with additional adapter proteins that hold it to the underside of the cell membrane. Once that interaction is broken up, the whole pathway tamps back down, Ras reverts back to its GDP-bound form, and you get the inhibition of phospho-ERK that we were seeing. So that hypothesis was verified when we created a cell line that had a mutated sauce in which the specific site of phosphorylation, the serine 1178, was removed and replaced with an alanine. And in that system, you see only Ras activation, you see only ERK activation, you never see kickover to an inhibition of ERK even with the most potent molecules. So again, we've done a lot of things with these activators. We've published quite extensively on it. The bottom line summary of this that you need to know is that several approaches, including serendipity, uncovered a diverse set of unrelated compounds that all do the same thing. They bind to sauce, they activate nucleotide exchange, and have consistent biphasic downstream signaling effects of RAS. And so before I end this unit on indirect inhibition of RAS, I just want to point out one last little thing. That is, while we were working on these activators, our colleagues at Boehringer Ingelheim were busy looking a little more intently for inhibitors. And they ran a high throughput screen specifically looking for inhibitors of the SOS K-Ras interaction. And they actually found a quite potent set of EGFR inhibitors in their compound library that did just that, acting as about 1 micromolar or so inhibitors right out of the gate. Now, when they co-crystallized these compounds with SOS, they found that they occupied the same activator pocket that we had been using. Here's the phenylalanine 890 that I've referred to over and over again. But they're occupying it quite differently, obviously. This phenyl ring sits in the hydrophobic floor. The quinazoline stacks with this histidine at the lip, and then projects this chemical matter right out into the space, right where RAS wants to be. That's the key, as they've published a little bit, that's the key to their so-called foot in the door to inhibition of this interaction. So their chemistry team did quite a lot of optimization on this. A 2-methyl group removed the EGFR activity. A lot of tweaking and structure-based design led to very potent inhibitors of the RAS-SOS interaction, including some that had very good PK suitable for looking in vivo. And look, they did. Again, much like some of the other molecules I've shown you, these compounds are capable of inhibiting phospho-ERK signaling in a number of different RAS mutant cell lines. They're also relatively potent inhibitors of cell proliferation in a 3D anchorage independent sense. And again, this is irrespective of which RAS mutation you have in the cell line. Even better, these compounds are active in vivo. Again, this tool molecule 3406 SOS inhibitor shows a nice dose-dependent tumor growth inhibition at a couple of different dosing levels. And even more so, it is incredibly effective in combination with other pathway inhibitors, namely MEK inhibition, as co-dosing with trabetinib shows. You can get sustained tumor regressions even with this combination. And lastly, I'll point out that in a very much related strain, BI has progressed the first known SOS1 inhibitor into the clinical trials for K-Ras tumors, providing potentially another option for patients that are suffering. So I've taken you very, very quickly now through a combined collaboration at Vanderbilt and Boehringer Ingelheim, where we've looked at both direct binding to RAS in its off and on state. We've looked at covalent inhibition of RAS. We've looked at binding to SOS to activate the system and cause paradoxical inactivation. And we've looked at direct inhibitors of SOS, more or less, if you will, an all-out assault on SOS and RAS right at the heart of the matter. And so what remains for me to thank are the people that have done the work. Again, there's a large cast of characters, most of which are represented here in the Physics Lab, and then an even larger cast that have worked on these projects in Austria, led by Daryl McConnell as the site head. And of course, as an academic lab, I do have to thank all the funding sources that keep us going. And so, Phil, I hope I have left enough time to entertain a few questions if there are any from the audience.
Yeah, that was wonderful, Alex, and thanks for that presentation, and great to see kind of working in collaboration with BI to take that through to the clinic here. So starting off with the G12C story first off, obviously you've come at that from a different angle to what Showcat and the others have done. What is the potency of the untagged warheads you've got on there? I mean, how much of it is driven by the affinity of just the ligand itself, right, and then the hot electrophile?
Well, so that's a big key. So I confess not to being able to talk really about the level of reversible binding, but I can state that it is our hypothesis that you should get some advantage in selectivity or specificity if you start with the sort of reversible binding approach. Now that's a theory, but as we've all seen, there's been some very nice work around the Mirati and Amgen molecules that have shown that those things are quite specific, really only sucking up RAS and I think one other protein when looked at. So effectively, is it going to be better? I'm not sure, but based in theory, our hypothesis was it should be.
And then pivoting across to SOS, I mean, you hinted at that this pocket may actually be naturally regulated. Have you got any ideas kind of what a natural ligand could be here?
Yeah, so that is a question that remains of fascinating interest to me and one that I do not have a good answer for. So it is my belief that nature doesn't put a pocket on a protein for no reason at all. And the fact that, especially in our work and even in the BI work, we didn't talk about this, but the so-called foot over here, it turns this molecule from an activator. So if you cut this off right here, this molecule, the rest of it is an activator. Putting this on, it becomes an inhibitor, indicating to me that that pocket really is, from so many different approaches coming at it, it's got to be a native control in some way, serving as some sort of feed-forward loop. Now, I can speculate about what it might be, but I've got no proof. I mean, SOS is a very large multi-domain protein, and the allosteric Ras GTP binding mode is already known to change the sort of auto-inhibited conformation of SOS, where it wraps back on itself. So I can imagine that that pocket that we have found also serves some sort of regulatory role with some other bit of the protein wrapping back on itself or not. But again, I have no proof of that and no experiments with results that I can show you. It is known that you can see this pocket in a few other exchange factors. So again, there may be something sort of conserved there across nature, but there's a lot of work yet to be done, I think, to understand the true biomolecular mechanism for why we get activation in this pocket.
Fascinating. And then just to kind of close things out and to kind of tie right the way back around to Lynn's talk at the start of the session, do you ever see any differences in kind of regulation of protein levels? I mean, do any of these activators or inhibitors actually degrade SOS?
So that's also an interesting question. I think in some ways that might be a response by the cell to begin to tamp down some of the signaling networks where it can in response to somebody perturbing it. One of the problems here, though, is that RAS is pretty critically important. If it goes away all the way, I think the cell is basically going to die. So there may be a blockage there on that type of degradation. Now that I know, people are probably working on RAS PROTACs as we speak, and so that question may come to a head later. But a lot of the earlier work that we were doing here, we weren't necessarily looking for degradation at that time. It was kind of before that became as fashionable. So I don't have a great answer, but it would be reasonable to expect that one of these might one day wind up being turned into a degrader.
Fantastic. Well, I just want to thank you, Rima and Lynn, here for some outstanding talks and also for keeping on time this morning. So you've set the day up really well here. For all of you on here, we're going to take a 15-minute break and then rejoin at 10:45 with the next session. So thanks again to our three presenters and look forward to re-engaging in 15 minutes.
And welcome for the next session, which is focused today on biotech startups. We're going to start off just talking about a project that I've been working on for the last year at the TMC, the Accelerator for Cancer Therapeutics. And then we're going to jump into a few of our amazing cohort projects and then finish out with a great startup in the community called Therapeutics. Just as a quick introduction, my name is Sarah Hine. I'm one of the entrepreneurs and residents with the TMC Accelerator for Cancer Therapeutics. I really want to thank the Gulf Coast Consortium for not only putting on the session today and allowing us to talk about it, but for really actually Suzanne Tomlinson and Stan Wadowich down at UTMB really incepted this entire program and helped submit the application that made it a real program today. It made this possible. And of course, the CPRIT, Cancer Prevention Research Institute of Texas, which supports this program and allows us to do the work that we're doing. So it's probably not a surprise to anybody today here, particularly if you've been paying attention to the conference thus far, that there are breakthrough cancer discoveries coming out of both the institutions here in the Texas Medical Center and really across the entire state of Texas every day. And it's really exceptional work that's been supported by the $1.9 billion of research funding by CPRIT over the last 10 years. However, what we see when we really dig into how does that get to the clinic? Sorry. Oh, man. How does that get to the clinic? What we see is that if you look at venture investment as just a marker for that process, because it helps drive commercialization and drive projects through clinical trials and ultimately to patients, what we see is that the Texas cancer research is significantly underinvested, not just in comparison to the hubs that we think of in California and Massachusetts, but even in many ways due to the enormous number of research dollars that we get in comparison to other parts of the country. And, it's not that this research doesn't end up getting out the door and to the clinic, it just has to make that significant jump many times to either the California or the Massachusetts ecosystems. And it makes it much more challenging for the next really innovative project to do the same. And so that's one of the challenges that we're trying to address here is, we really sitting down and thinking about, if you look at that process of therapeutic drug development, the very early research is typically supported by academic research grants. That could be through CPRIT, that could be through the National Cancer Institute, or it could be through, say, a foundation. And that usually takes you through maybe early preclinical development through some proof of concept studies. But then as you start thinking about going later into the clinic and really testing that technology on patients, what you see is that not only does it require significant capital investment to support that project, the skill set and the expertise that's really required to make that transition shifts dramatically. And so a lot of times, the way people address that challenge is through maybe forming a company and trying to establish sort of a new team to take that project to the next level. The challenge there is that, this is a substantial effort just to even get this off the ground, find the right people, and develop that strategy that helps make it credible to drive forward. And so it represents a pretty significant barrier, and there are many projects that don't manage to overcome that barrier for a variety of reasons. So that's really the challenge that we're trying to address here, is to help accelerate that path through those formation stages. And the way we view it is it really starts with the people that you can bring together. And so the program, although it has many features as far as curriculum goes, and other resources we're going to talk about, it's really about bringing together the people, the founders, bring them together with talent and knowledge, and helping them understand what it is that's going to have to be done with their individual projects, and therefore creating a solid foundation. The formation of a company is not typically talked about in the press releases. It's before the big funding, it's before you get the big names behind it, but it's a really critical stage that enables all of that to happen, and doing it right can really be the difference between years of effort. And so when we figured out where are we going to do this, of course we thought that there might not be a better place to do it than here in the Texas Medical Center, where we're surrounded by a community of really great researchers and institutions that we can help collaborate with, and gain that expertise at the cutting edge of this field. And that itself is sort of based within this larger community across the state that's was already at the forefront, but has been really just taken to the next level over the last 10 years. I have the privilege of being able to talk to you about this today, but it's really been the efforts of the people that you see here, as well as a very substantive advisor network, and a lot of people really just pulling in the same direction, seeing this vision and going after it and figuring out what does it really look like, how do we meet these challenges, and how do we bring people together, get them excited, and really help drive these formations. We're also supported by a truly exceptional executive advisory committee. We're really fortunate to have the time and commitment of everybody on the slide. We have representation of the venture perspective, signed people who have gone through that early development process themselves, seen those challenges, seen the challenges of taking projects into the clinic, and really of recruiting the right team members and understanding how do you bring the right people together and create a new entity to move forward with. And with that team that you saw, both the Executive Advisory Committee, our internal team, and really our advisor network, as we were reaching out, talking to people, we came up with a couple core tenets that we sort of call our guide star. And that's, again, really just giving people the access to the talent and advisors and consultants that they're going to need to build a really credible plan to move forward. One of the things that I've certainly experienced myself as an entrepreneur is you're overwhelmed with the amount of people, and it feels very inaccessible. So just having a introduction to the right person can sometimes really make the difference between sort of not understanding where to go and sort of having that clarifying experience. Supporting product development both through the computational chemistry resources that we offer and access to the secret core network. And ultimately, our goal is to help these companies be viable, and that's through taking those plans that we're sort of putting together during the program, and hopefully that can be distilled back into both non-dilutive grant applications, which we do directly support, and giving people the confidence to understand as they go into venture capital raises what it is that they're bringing to the table, what are the challenges, and what is that path forward. And so they can have a really high quality discussion that can build a lot of confidence in their program and ultimately hopefully lead to a successful funding effort. The cancer therapeutics development plan that we're helping people put together is sort of through a number of things. It's really just understanding what does that path look like, and we support that through a large amount of curriculum, both sort of outlining both the company formation side as well as the and then interlacing that with the product development side. Really just helping people understand, working through what are the timelines and the costs associated, and then more importantly almost, are there any critical gaps or key experiments that you can do to either make people more comfortable with the inherent risks of drug development, or really build a lot of excitement to help drive that project forward. And so at the end of the program, what you should have is you sort of have a really good idea of what stage are you at, what do you need to do, and what are your key go, no-go decision points to not just achieve funding now, but when you get that funding, to drive through the milestones that'll get you to the next round of funding. And so you're planning, several steps ahead and able to move more quickly through the process. One of the really unique components of this program is that we're able to incorporate both actually outside of the accelerator to the community at large, a computational chemistry resource, which, as I'm sure you've heard over the last couple of days, has really just revolutionized the way we think about small molecule development, but is often inaccessible, particularly if you're more of a biologist than a chemist, but you've been working on biological processes and have a deep understanding of what could be possible if you could only create the right targeted molecule. And so this is available at no cost. If you're in that position, I would recommend that you reach out to us, and we can connect you through to Amit, and he'll work with you to generate some leads. You can take that back to the lab, and we'll be communicating with you as the EIRs and helping you sort of think about what does this look like as a comprehensive plan. And then hopefully in the future, if you start to see successes, you'll have an understanding of whether or not you want to drive that forward, and we could support you through the accelerator as well. The secret core network, I think you've seen speakers from a number of these cores over the last day or two. Is really just an amazing set of resources that have been built up through the efforts of the people that I've already outlined. And so the great thing about this is it's really a network. And so what we can do is we can help you plug in with these resources and access, maybe at a reduced cost, key de-risking or product promoting research that will help you promote your project during the program. And sometimes, for example, understanding your PK/PD can be really critical in those discussions that you have and understanding the viability of your project. Over the last year, when I started, we were really still in the planning stages. We were trying to understand what it's going to take to take this vision and really detail out what the execution of that vision could look like, and then go search and talk to people and share this vision and see what their needs are in response to that. And, that problem statement I outlined sort of at the beginning of this conversation it's really a hypothesis, right, of what is the challenge that people are seeing, and is there a way to address it? And I got to say, we were just overwhelmed by the quality and quantity of the research that we were seeing as we really went out and told that story and, started trying to understand if, how to help people and would they be willing to come along this journey with us. And so, we started with this year in January with 15 really amazing projects and companies, or if they weren't companies at the start, they were rapidly becoming them. And I asked Suzanne if I could talk for 2 hours and really just go through every single one and why I think they're really amazing. And unfortunately, she told me that I wasn't allowed to do that. So instead, what we're going to do is, highlight 3 of these projects and have the founders speak to you about sort of what they're doing, why we thought it was really interesting and where they're going with it. And I, hope that you'll be as excited as I am. What we found is that, I think people are really willing to engage with this process. And it's really just about giving them the tools that, scientists are problem solvers at heart. And if you just give them the tools, they are more than willing to pick up the banner and drive forward with it. So it's been a really gratifying experience so far. I'd love to answer any questions. I think you're allowed to drop them into the chat if you would like. Otherwise, what I would say is that you should reach out to us really at any time. Ashley Francis is our amazing program manager. She will be more than happy to help connect you to the right resources, either us as entrepreneurs and residents, Amit Gupta, who is our computational chemist, or even the other resources within the TMC. And this is, if you're just even thinking about, you think you're working on something, you're trying to figure out what to do with it, maybe it's a company, maybe it's not, maybe you can access some internal resources at your institution. We're here to help you find the best path, because that's our goal, is to get this research out the door and to the clinic and ultimately to patients.
We're going to bring up Jason Bach from MD Anderson. Dr. Bach has spent the last 20 years, maybe more, before moving to MD Anderson, really working in several biotech and biopharma companies focused on developing biologics. And during this 20 years, it's just incredible, I think anyways, that he's moved at least 15, probably more than that, drugs through preclinical development into clinical studies, which is a major accomplishment. But even more so, 3 of those now, as far as I understand, are actually commercially available. So it was a great coup to MD Anderson to entice Jason to come to Houston. We're delighted to have him here, where he now is a VP at MD Anderson and heads up the biologics product development. So looking forward to his talk and understanding how he's doing this. So Jason.
Stan, thank you so much. You can hear me okay?
Yes, indeed.
Okay, great. Thank you for the kind introduction. Thank you to the organizers for inviting me. I think it's perfect, the setup. I guess the organizers are pretty smart about having me follow from Tom and Cleona. I'm going to sort of straddle the line between their presentations, both talking about, I think what Tom said was his #1 area for advancement in cancer, which was immunotherapy and specifically cell therapies. And then we heard a great example of that from Cleona just now. Just to give you a little perspective on where I've come from, Stan gave that kind introduction. I started my career in academia, undergraduate at MIT, where I actually many years ago, before cancer immunotherapy was a thing, I was in Richard Mulligan's lab studying cancer immunotherapy. Then went on to Stanford, working in Richard Scheller's lab, where the lab was basically involved in finding new genes. This is before the genome was sequenced, and figuring out what they did. Then from there, I think as Tom said, I, to be honest, wasn't interested in writing R01 grants, and the field of sequencing the human genome was just exploding. It seemed like a good company to join was a company called Human Genome Sciences. This is in Rockville, Maryland, and a company focused on tapping into this very new treasure trove of new genes and trying to figure out what they did and then turn those genes into therapeutics. And so HGS had quite a big engine of finding new genes and then screening them for ones that could be good therapies and taking them into the clinic. And so I really learned how to develop things preclinically and file INDs and get them started. This was about a 1,000-person company when I joined. Then after 5 years, a group of us formed a friendly spinoff from HGS called Cogenesis, and about 50 of us left and formed a startup, biotech, just down the road, where we were focused on a subset of HGS's intellectual property and taking those products through preclinical stages. And it was quite an intense period. Within the first year after we spun off, we brought 3 novel drugs into the clinic and started clinical testing of those. As we were developing these products, we were having various partnerships with a number of companies. One of those was Teva Pharmaceuticals, who said, well, it looks like you have some really interesting products here, but we are, as Teva, as a company, is interested in getting a foothold into innovative biologics. And so rather than partnering with you on one particular product, we'll buy the company. So our little startup lasted a total of 18 months and then was acquired by Teva Pharmaceuticals and became the innovative biologics engine within Teva, which was a 50,000-person large biopharma, primarily focused actually on small molecule generics, but we represented this innovative biologics area. During 10 years at Teva, it was a fantastic experience where we not only built up quite a big infrastructure, we grew that organization from the 50 people we started with to over 600. We created an R&D, innovative R&D site in Westchester, Pennsylvania. And with that site, we were able to bring 15 new products into clinical testing. And eventually 4 of those we brought to commercialization. And as Tom rightly said, it is amazing when you get to commercialize a product and just see the scale and power of large biopharma to be able to bring a product to tens, if not hundreds of thousands of patients on a really large scale. So it was bumping along as a fantastic career. I was site head of the 600-person site. And then some of the folks, Julio Draeta, Phil Jones at MD Anderson started talking to me and really intrigued me with the idea of coming to MD Anderson and working in cancer immunotherapy and really driving biologics product development forward. And so I'm going to spend the rest of the presentation talking through a little bit over the last 2 years, what we've done, how we're thinking about it, how we're approaching. I think, I've been fortunate and had quite a bit of success at each one of these different organizations. And it's been to be honest, it's been a different solution at each place. And so it's important to, I think, take the time when you come into a new organization to figure out what are the strengths, what are the, if you want to call it, competitive advantages of that organization, and then what can you do to enhance those and to really make an impact. So let me tell you a little bit about how we're thinking about that at MD Anderson. So first, and this goes along with what Tom said about the exciting new areas. I think if you think about the big buckets in medicine over the last 100, maybe 200 years, this is a good example of those large buckets. All the way from sanitized water to, even a few years ago, we had sort of taken vaccines for granted. I don't think over the last year anyone's taking vaccines for granted anymore, how impactful they can be. And then small molecules, and then especially protein therapeutics and monoclonal antibodies. And I think we're, while it's been building for a number of decades, we're at a really exciting point with these living drugs, cell and gene therapy. And as you guys know, you can't, read any headlines in biotech or pharma these days without some exciting new cell therapy innovation or clinical response reading about that. So it's a hugely impactful area. And MD Anderson has been involved in cell therapy for decades, whether it was on the clinical side with running large phase 3 clinical trials, the key studies for Yescarta. More clinical trials are run by MD Anderson Clinical Center than any other cancer center in the world. So it's a pretty amazing nexus for running cell therapy trials. And in addition, has a large research engine called sort of bidirectional translation to both come up with novel ideas and concepts, much like Cleona described, and taking those into the clinic and figuring out the efficacy in the clinic, as well as then once you're in the clinic with those, understanding how that response is being carried out, and really figuring out why patients respond or don't respond, and interrogating that. And so these have been the two pillars where MD Anderson has been a leader in cell therapy for quite a bit of time. And here's a concept I want to introduce to you that's a little bit more about product development. And I'll show you a couple of these curves where on the y-axis is value of a product and on the x is time. And so I think intuitively we understand that as we're developing a therapeutic, as we understand more and more about it, as we prove that it works, let's say in vitro and then in mouse models, and that the value increases. And in academics, we focus a lot on these preclinical models. In the big scheme of things, there is some value that is overall created at the preclinical stage. It can take a long time. This research phase can be one of the longest phases and one of the areas of highest attrition. But then there's a really a sharp increase in value in products as you take them to IND and start generating clinical data and reach clinical proof of concept, and then eventually commercialization. And this is sort of due to, well, a number of factors. Because oftentimes the clinic can be difficult to reach from academic centers, oftentimes products are partnered at preclinical stages. And I think just like Cleona showed, there can be a lot of value gained and a lot of insights gained by taking products into the clinic and understanding how well they work. And so, as well as getting those closer to eventual commercialization and increasing their value. Now, so, you can see why there's some advantages of trying to bring products further into clinical development and closer to commercialization, but there's also some challenges. As excited as we are about the potential of cell therapies and how much, it's amazing how much capital is pouring into these therapeutic modalities. I think when you look back at especially 2020, it was a pretty rough year for cell therapy and the FDA. There were a number of delays, rejections, what's called complete response, where you're trying to commercialize your product and FDA says you need to do some more work and come back later. for major companies from BMS and Juno to Iovance and others. That really, I think, is a wake-up call for the cell therapy area, and that all of these companies showed quite impressive clinical results, and so carried their products into the clinic, showed great clinical results, but all of them ran into roadblocks on the CMC or the manufacturing portions. And I think what FDA is clearly sending a message to the whole community of saying that amazing clinical results are fantastic. We will work with you. We will give you breakthrough designation and try and help you get those products to patients as quickly as we can. But there's not going to be any more cutting corners in terms of having robust manufacturing processes and the like. And so, you know, it's up to us now how we respond to that. And so, you know, just going back to this curve, you know, it's, you know, we create all this value getting through clinical proof of concept. But then if we get stuck where we're delayed at being able to commercialize these products due to manufacturing issues and FDA requirements, you spend a long time without creating any value. You know, we know the product works. We just have to figure out a manufacturing process to make it robustly, or a release assay, or various different, we need to do comparability studies to show that the product we're currently making is the same as the product we've historically made. All of those things don't, to be honest, create much value for the product. We already know it works. We just need to know how to make it. And so we really want to avoid getting stuck where we have a very valuable product that can help a lot of patients, but we can't get it to them for manufacturing issues. And so the new paradigm that have been setting up at MD Anderson takes this into account, and that taking the two pillars of historical strength of the clinical cancer hospital and the translational sciences, but marrying those together with a group experienced with drug development and understanding FDA, as well as industrial manufacturing facility and capabilities. And this was sort of foretold a number of years ago by Scott Gottlieb, you know, before he was on, you know, all the morning talk shows and everything talking about COVID. He was commissioner of the FDA. And what he said is, you know, for traditional drugs, FDA would spend 80% of their time on the clinical, the risk-benefit analysis, adverse events, efficacy, and maybe only 20% reviewing the product. This is for small molecules and maybe protein therapeutics. But now for these cell therapies and for these gene therapies, they're spending 80%. FDA is spending 80% of their time and most of the problems on the manufacturing side and the product development side. And so I think it's incumbent upon us as an industry or as an ecosystem to respond to that. And no longer can we just say, well, look, as long as we see some clinical responses, we'll figure out the manufacturing later. That clearly, I think, is not working very well for us. And so we feel that having this integrated approach between all four of these aspects really will allow us to have the maximal impact. And so how did we approach this? First, in terms of manufacturing, a facility is critically important. Being able to control the environment and have consistency in your environment allows you to produce consistent products. And so we were fortunate a few years ago that a biotech company called Bellicum was interested in selling their facility, and we acquired that facility, which is literally 2 blocks down the road from MD Anderson's main hospital. So we acquired this 60,000 square foot cell therapy development and manufacturing facility, has 14 wonderful clean rooms, as well as all the supporting areas to support GMP manufacture, as well as suites to produce viral vectors. The facility construction was only completed a few years ago. And some people transferred over with the facility, but we've been busy over the last year hiring and building up the staff. So, that started by hiring a senior leadership team of people who had a diversity of experiences, some experience in academics, but the majority of experience in industry, developing products, understanding what are the important aspects of product development, CMC, manufacturing, and FDA regulations. So it's a fantastic team we've pulled together over the last year. I'm really humbled to be working with them. In addition, we've grown the overall organization from a year ago. It was about 6 people to over 60, which is pretty staggering growth. We are trying to, as some of the earlier speakers mentioned, really put together a diverse team that marries together such strong academic science, which is critical, I think, in this cell therapy area, which is depth of understanding of biology and immunology, as well as mixing them together with people experienced in industrial drug development. Just as a side note, we have a number of other positions open, so if anyone's interested, please look at our website, and we'd be interested to talk to you as we continue to grow this, because really it's going to be the people that drive the difference going forward. Okay, so I've told you a little bit about our model to really come up with an impactful way of developing cell therapies. I'm a big believer in engineering solutions. So like seeing what you want to do, coming up with an intentional plan on how you're rigorously going to approach that, put those elements in place, bring in the best people, give them access to the right equipment, the right supplies and things, and then allow this to flourish. So what we think by putting together this model is, yeah, there it comes. What we think is that taking our standard curve, that a value curve, what we've talked about, that with this model, we're able to steepen this value inflection curve by decreasing the time it takes to bring products into the clinic and shortening the time once they get into the clinic that they'll be ready for commercialization. Reducing the manufacturing cost and risk associated for these products that are fairly complicated. Having to weave together a network of CMOs can be very complicated. If you need one for the viral vector and one for the cell therapy, and you're coordinating an autologous therapy and shipping PBMCs around to these various CMOs, it can be very complicated. We can do that all in-house. And if by having an industrial approach to these products right from the beginning, we hope to avoid this stalling of the product after clinical proof of concept, because we'll already have industrialized the process and be ready to seamlessly move to commercialization. As we put together this engine, there were so many innovations at MD Anderson in the cell therapy space across the modality landscape, whether it was allogeneic therapies, which may require, as Clearona nicely showed, more engineering and more thought about how to design them, but then have such potential to be off the shelf, to, as you move across the slide from left to right, CAR-Ts that are more costly to manufacture and need more adjacencies between the manufacturing and the clinic. While that's definitely a challenge for commercialization, at MD Anderson, we're well positioned as to try to develop also these autologous therapies. And so across this spectrum of different products, we're able to participate in various, we have different products in each of these different platforms, if you will. And I think why we think that's important, we can go into a whole nother talk, but there's going to be synergies across these. And, a challenge in biotech or other organizations is just, having to focus on one particular class of these. We think there's going to be some synergies that we're going to realize across these different platforms. In addition to being able to take MD Anderson sort of invented or discovered therapies, cell therapies, and industrialize and take them through proof of concept and have them ready for commercialization. As we put this model together, there were also a lot of interest from biotech. And so about 6 months ago, we signed a partnership with Obsidian Therapeutics in Cambridge to take their IP, their technology for regulated expression of cytokines and marry that together with MD Anderson's experience in TIL therapy. And that together will co-develop a product with regulated membrane-bound IL-15 expression in TIL and take that through clinical proof of concept. It's a really relatively novel strategic initiative where a biotech company is coming to an academic medical center, and together we are developing a product and sharing both sharing in the upside opportunities that may come from that, and deciding on the regulatory strategy together, deciding on the clinical strategy, and really in all senses co-developing that product. So finally, I want to sort of zoom out and try and actually this sort of touches back to what Tom was talking about the overall ecosystem for drug discovery, development, commercialization. As he so correctly said, we know that the innovation occurs in academic research labs. And that in some ways, the way of what he said in terms of the product development, turning those innovations into developable products really happens in startup and biotech. And then the commercialization of those, or the final development and then commercialization of those, really happens with the scale of large pharma. The challenge with this ecosystem that has worked, quite well, is it requires a lot of handoffs along the way. You can see that from all these arrows, including, the clinic and the patient. And so with these very complex therapies, with the complex manufacturing and CMC that goes along with it, we think one of the reasons for the challenges currently in cell therapy manufacturing is because things keep getting handed off along the way, and no one ever is able to really dig in and see things through to the finish line. By the time large pharma gets their hands on the product definition is already there and restricted. And so, our approach is to combine this, the innovation in academic research, the clinical capabilities, and then the industrialization capabilities all together within MD Anderson to develop cell therapies through to proof of concept and be ready to commercialize them, likely in partnership with large pharma. Of course, we always have to remember that the end goal of this is giving these novel therapies that we have incredible efficacy, a chance to have the largest impact on patients that we can. So thank you for your time.
Jason, wonderful as always. Thank you very much. I do have one quick question, and just in the interest of time. So could you comment on what you view as some of the limitations now of translating or expanding the distribution of these cell-based therapies from a resource-rich environment like we have in the medical center in the US into perhaps more resource-limited environments around the world? What are those challenges, and how do you see overcoming those challenges? I'm curious.
Yeah, that's a great question. There's a lot of aspects to that. overall, as we understand these products better, we will need to be able to develop more robust and more simple manufacturing processes that are more enclosed and require less complicated human intervention and more automation. As we understand them better, we'll understand what are the aspects that are important to control, what may not be important to control, and that'll allow us to simplify these and expand their reach. So I think the major cancer centers will have a role, because you can't just wing this and say, well, let's just make it simple. Actually, you have to understand in a great deal of depth first. Once you really understand, then you can simplify. And that's our goal, to be able to expand the impact.
So do you think you'll be adding, as you expand your network, some research capabilities into kind of the manufacturing process on how to, or do you think that'll be other groups getting involved with you, or a combination?
I think exactly like you said, a combination. We've created this engine, and we're very open and interested in partnering it with various different players. So we've talked with a number, there's, as you can imagine, capitalist system, there's a demand for like these simplified, we call them manufacturing in a box systems, where you have a box, you put PBMCs in one side, and out comes a CAR-T on the other side. That's the dream. There's a number of companies developing these, and we're not going to try and invent that ourselves, but we will partner with them, use our strengths in being able to develop products and carry that into the MD Anderson clinic to help those companies achieve a proof of concept that their product can work in the lab, but can work in a clinical setting as well.
Great. Well, thank you very much once again, Jason. Delighted to hear the progress. So I thank all the speakers that have joined us today. I thank all the participants who have listened, who have gone to the poster sessions, who have participated with the core groups and the breakout sessions. And hopefully you've learned something. Hopefully you're inspired. Hopefully we see some new companies coming out of some of the faculty, postdocs, grad students that have joined us. So with that, I'll say good night, and everyone stay safe. Bye for now.